Dr. Vijay Shankar Raman is a botanist, senior research scientist, National Center for Natural Products Research, co-director, University of Mississippi Scanning Electron Microscopy, core. His areas of expertise goes to plant identification, botany, plant taxonomy, morphology, anatomy, ethnobotany, medicinal plants, herbal supplements, microscopy, preparation and management of herbarium, development and management of respiratory of botanicals, GMP and quality control of botanicals, floristic surveys, threat assessment and red listing, medicinal plant conservation. And he has got 20 years of experience, and he published more than 125 research publications, authored 4 books, and also 42 conference presentations. And he has 7 camp reports and papers revised more than 100. Reviewer for journals 32. He's a member of editorial board in 80 journals. His ResearchGate score is 33.5, citations more than 1,000. He's a member of life member of associate member of American Society of Pharmacognosy, life member of Indian Association for Angiosperm Taxonomy, India, member of the Botanical Society of Japan, member of medicinal plant specialist group of the IUCN's Species Survival Commission, moderator and pillar of eFlora of India Google group. And is a reviewer and editorial board member of many journals. And he is a senior research scientist yeah, this is many more things are there, and if you read out, we consume the time. And once while I contacted Dr. Vijay Shankar Raman sir, immediately responded, and he agreed to be a resource person for one of the session. And thank you, sir, once again. Thank you and welcome to the session. And on behalf of again our organizing committee members, Dr. Shashikala and Dr. Nageshwara Amanchi, we welcome you, sir. Thank you. You can start, sir.
Thank you so much, Dr. Vijay Bhaskar Reddy. First of all, I would like, can you hear me?
Yes, sir.
Am I audible?
Yes, sir.
Okay. So first of all, I would like to congratulate the organizing committee, Dr. Vijay Bhaskar Reddy, Dr. Nageshwara Amanchi, Dr. Shashikala for organizing this important national seminar on plant taxonomy. This is very important and it's very timely, you know. So botanists are like endangered species. So we need more botanists. We need more trained botanists. So that said, I would like to, I'm going to share my screen. Can you all see the screen?
Yes, sir.
Yes.
Great. So today I would like to discuss with you all about authentication and quality control of botanicals. So as you know, there are different types of authentication methods. There is a physical method, chemical method, and molecular method. So there are a vast number of analytical methods are available for authentication of botanicals. So today we will focus on the macro and the microscopic approach. So when I say botanicals, that includes medicinal plants, herbal plants, and natural, it's also part of natural products. Medicinal plants, there are different, like, you know, the usage is different. How the botanicals are regulated, it differs from country to country. So in India, we use like, you know, we have traditional systems of medicine, but these days the dietary supplements are becoming more and more popular. So anybody can buy botanicals from online sources and supermarkets like that. We all know the importance of medicine plants. There is no need of introduction. Like more than 80% of the population in developing countries, they still depend on medicinal plants for their primary health care. And there are some statistics in this slide, so you can see also, this looks like COVID map, right? The COVID-19 map. No, this is the how the botanicals are used annually, like how much botanicals are traded annually. This is the market trend. As you can see, every year the demand and supply for medicinal plants has been on the rise. So there are many reasons why the botanicals are getting more and more popular. There is a wide acceptance for traditional systems of medicine and the natural products and botanicals because of their safety, efficacy, and also the traditional systems are thousands of years old. They are time tested. So there is a wider acceptance for medicinal plants. In India, we have around 8,000 species of medicinal plants. Almost 50% of the plant species are used as medicine. So this growing industry, this is a very important, and it's encouraging information, that is the usage and demand for medicinal plants is on the rise. But there is a problem in the dietary supplement industry, in the botanical industry, because there is no global level control system. There is no regulation controlling the entire botanical sector, dietary supplement sector. Of course, there are guidances, there are certain rules, but there is no standardized system for the entire industry. This global botanical industry is threatened mainly by the practices such as adulteration, contamination, and substituting medicinal plants. Adulteration. So there are, when you see, there are so many reports coming, being published because of, noting the problems using medicinal plants. Like there are some side effects and adverse reactions. But when you see, there are researchers, they show that these kind of adverse reaction happens mainly because of using adulterated botanical. Okay, so our traditional systems are time tested, more than 5,000 years old. But if there is something happening using those medicinal plants, it means that there is something wrong with the material being used, right? So the toxicity and the adverse reaction in consumer health problems could be, mainly because of adulterated botanicals. There may be problems when someone is taking herbal plants, and if they are also taking other type of medicines like modern medicine, allopathic medicine, there may be interaction, drug-drug interaction, botanical drug interaction. But most of these adverse reactions are happening because of using adulterated or incorrect botanicals. So as I said, adulteration has been, it's again, when the commerce started, time immemorial, at the time the adulteration also started. So it's a very old history, but still happening. Now it is getting more and more advanced because of the advanced technology. So adulteration also becoming a major challenge in the botanical dietary supplement industry. This is like coronavirus, how it is affecting the human health and the humankind. Like adulteration is a huge problem in the dietary supplement industry, in the botanical industry. So adulteration happens in multiple ways. Like it can be unintentional. It's not that all the people who are involved in the botanical dietary supplement industry are botanists. There are so many non-botanists also involved, and there are very good industries. They follow the GMP, GAP, all these guidelines, but not all. So adulteration happens because of the misidentification, careless collection, and not knowing what is the correct plant, what is the substitute, what is the wrong plant. They just collect everything, whatever they think it is a plant. We will come to some examples. And also intentional adulteration, that is like fraudulent substitution or addition of substances in the product. So this is the most dangerous. This is also a dangerous adulteration. So just because they want to get more money in a very short time, they just add things to the botanical products. So we will focus on some of the areas of unintentional adulteration in this presentation, like how we can avoid a misidentification, how to establish authenticity of the botanicals, all these. So in order to do authentication, so we need to have basic understanding of the plant morphology, taxonomy, botanical nomenclature, herbarium collection. All these tools are necessary. Although we don't need any hi-fi equipment, hi-fi instrument to do plant identification, but we need trained personnel, experts to do the authentication process. Herbarium is one of the major tool that is needed for plant authentication. And some of the challenges could be having multiple common names and a long list of synonyms. But botanists, this cannot be a problem for the botanists generally. But the dietary supplement sector, the botanical supplement sector has a number of non-botanists. So for them, it may be a huge problem. For instance, Central Central Asiatic has more than 40 synonyms, and Diascorea has almost 30 synonyms. There is, here, we all know what comes, why we have multiple names for a single plant. As botanists, we know. But for non-botanists, it's a big problem. It's a challenge. Again, common names. Common names are very useful, and even non-botanists can identify plant based on the common name. It's easy to memorize. But there may be multiple names for the same plant, and sometimes same name is shared with other plant species, multiple plant species. For instance, fig, the name fig is applied to multiple ficus species. Similarly, pinus, pine tree, different genera, different species of gymnosperm called as pines generally. In this case here, you see Missouri snake root. Here, the actual Missouri snake root is supposed to be Echinacea purpurea, but for a long time, Parthenium integrifolium has been used and considered as Missouri snake root. Even some of the research publications, they used Parthenium. It's a wrong plant because these plants share the same common name. Okay. So in this example, you can see all different species of plants. They all known by the same common name. In this case here, elephant ear plant. So it is applied to Alocasia, Philodendron, Acacia dani. They all belong to different family, different genus. As you can see, just because they share the same morphological feature, they got the same common name, elephant ear plant. And sometimes it may not be, like in this case, Hemantha albiflos. It doesn't even look like elephant ear plant, right? It should be called as elephant tongue plant instead. So common name can be confusing. In this case, the name bay leaf is applied to multiple species, belong to different genus, different genera, different species, sometimes Lariaceae, Myrtaceae, different families also. Because they are all aromatic plants, they are used for the similar purpose, same purpose. They are all called as bay leaf. But actually, the Laurus nobilis, that is the true bay leaf. I'm not sure if you can see the mouse moving. So this is the Mediterranean bay leaf, the true bay leaf or sweet bay leaf, But there are multiple other plants like Californian bay leaf, West Indies bay leaf, Indonesian bay leaf, Chinese bay leaf, Indian bay leaf. For most of the Indians, cinnamon is the bay leaf. So because it is commonly available in India, so cinnamon tamala is used as bay leaf. They all have this, they're all aromatic plants. They can be used, but they are different in their chemistry, their properties are different, and even their flavors are different. So you should know what bay leaf you are using, for instance. So for authentication, as I said, botanical nomenclature is a must. You should know the basic botanical nomenclature because the labels can be misleading. So in this case here, so there is, it cannot, the label many times does not tell what is there in the box, in the package, or in the product. So it could be misleading. So in the US FDA has put out some rules that all the botanical products, the dietary supplement products, they have to follow these rules for making labels. The label should contain the specific name of the plant, for example. So you should not just give the common name as the product name. So it should have, even if you use the common name, you should have the botanical species name, the complete name. And the name should follow the international standards of botanical nomenclature, like ICN. So I'm not going into details of the botanical nomenclature. You have listened to lectures from experts on nomenclature, so there is no need to repeat. So here in this product, there is an example product. It says bay leaves. As I said, there are at least more than six species are used as bay leaf and also traded as bay leaf. So one should know what species we are using. So that's why the label should provide the complete information. So if you see here, you cannot see the scientific name of the product, right? What is included in the product. So we used to, during training programs, we used to tell the participants how the label should be prepared, what information should go in, to make things clear. And sometimes misidentification, it's not sometimes, misidentification is very, very, very common on the internet or in different places. And many of the products are also mislabeled. So in this case here, they say it is Laurus nobilis. This is a screenshot, but it is actually Prunus laurus erasus. Okay, there are some Prunus species which are poisonous. So we don't know what species they're actually using in the product. Another important example for confusion with common names. In this case, this is a traditional Chinese medicine, fungi. Okay, the actual plant is Stephania tetrandra. This is the plant used in traditional Chinese medicine. But the name fungi is also applied to other different species like Aristolochia fungi and Cocculus orbiculatus for some reason, maybe for their similar appearance or their distribution or use. It's not usage in this case. What happened? One of the batches of products, they used a wrong plant, Aristolochia fungi, because the people who collected the plant, they collected Aristolochia instead of Stephania because they were told only the local name. They did not know the botanical names. Okay, so they collected Aristolochia fungi, which is known hepatotoxic. Aristolochic acid is present in this plant. It's highly toxic, and those who consumed the product, around 30 people who lost their kidneys because of using the wrong material. So common name can be fatal sometimes, going with the common name. So even if you are collecting a plant, you can refer to common names, but make sure to ensure the correct identity scientifically. So many authors, many researchers have discussed and wrote articles about the importance of botanical nomenclature, the scientific names. So there are many papers dealing with the importance of nomenclature. Here are some examples. So Rivera et al., they just checked how many scientific papers use botanical names and how many are used in a correct way. So according to this study, they analyzed, they verified 214 journals, 214 articles from Journal of Ethnopharmacology and 214 articles from the journal Phytomedicine. These are all very popular and highly reputed journals. So all these articles, they use the scientific names of plants, but as you can see, a large proportion of the papers they have used in the wrong way. So the botanical names are used incorrectly. So using incorrect botanical names, in the next slide, I'll show you what are the different types of errors people make. It makes the paper, it loses the value, because if the name is not correct, first of all, if someone tried to search a paper with a botanical name, if the paper has used a wrong spelling, you won't get in the search. Okay, and also it misleads, and if the name is incorrect, and if the raw material is not identified correctly, so it loses the value, and it's not going to help anyone. So that's why the scientific names in the publications should be accurate. So as listed here, there are so many different types of errors. You can see even in botanical papers and the papers published in reputed journals. Imagine there are so many hundreds and thousands of predatory journals, like a mushroom. They are coming up and publishing any type of articles. You can imagine how many errors you will find. So all these, it's like a junk literature. So there is no use in having incorrectly identified study materials and incorrect botanical names. So of course, anyone who is working with the plants, they should know the basics of nomenclature. As I said, I'm not going into details of this. You have already listened to best lectures. So the problem is, as I said, there are so many non-botanists who are also involved in the botanical sector. So they should be properly trained, and they should know the basics of nomenclature. So as botanists, we know why the synonym comes, and whether it is from the same herbarium specimen, same type specimen, or different types. There are many reasons. And also author name standardization. So we all know that there is a standardized abbreviation of author names of botanical names. But I will show some example. So not everyone is following this. If you're using a botanical name, if the author name is not included, or if incorrect author name is added, it completely changes. So name is everything. Like Shakespeare said, what's in a name? Rose is a rose, right? That's okay. But here, when it comes to botanical sector, name is everything. The name should be correct. It should be accurate. Only then you can link whether it is a product or literature. So only then you can link properly. So basically, training on taxonomy and nomenclature and herbarium techniques can help fix the problem. And as a botanist, you should know the basics of all these tools, And when you write um, research articles, make sure that your raw material is correctly identified. And so here are some suggestions for the journals. Any journal that deals with papers related to botanicals, the papers have to be verified by a taxonomist, or the reviewer or the editor. At least the editor should check whether the nomenclature is correct, right? Whether the latest name is followed, whether the supporting material is there, herbarium voucher specimen is made and properly identified and deposited in a herbarium. So all these things can make the paper more valuable and more accurate. I'm sure you all know there are several authentic sources in the websites you can check, and there are floras for verification to get the accepted name. So when you are writing articles, you should follow the latest accepted name and with the proper author citation. Okay, and there are so many websites available to help, but you should be careful. There are also hundreds and hundreds of websites. They provide incorrect information. Okay, so you should know what to refer and which website to refer. So here I have given some suggestions. I'm sure you all know this. Plants of the World Online. I used to check this website because this is one of the most advanced, one of the latest and actively updated websites, and it covers a larger proportion of plants from across the globe. There are other websites, like if you are working on medicinal plant, you should refer to this website, Medicinal Plant Name Services. It provides you with the accepted name and the common names along with the references. So which reference is citing using which common name. In addition, there are so many other helpful information you can get from these sites. The plant list, this is the very commonly used website for botanical name and synonymy. This is very, very helpful website, but it is not up to date. So they update like a batch by batch, and it may not be up to date, So that's why you have to refer to multiple sources along with the local floras. So this one will give you the synonyms and the accepted name, but you have to check whether it is the latest. IPNI, this is one of the standard websites sources you can check for getting a lot of information about when and where the botanical name was published first, like protologue information. But this doesn't give you what is accepted name and what is synonym. But you can get the correct abbreviation of author names and also the original publication details from IPNI. Of course, for authentication, after collecting the plant, you will be checking with the local, regional, and national floras. And there are many online floras available, e-floras. This is just an example. We have e-flora of China, e-flora of Pakistan, North America. There are many countries have complete e-floras. And there are many other authentic sites. They will provide you, they provide authentic and detailed information. And if you're working on Asteraceae plants, better check with this website, Global Composite Checklist. And this is for legumes. And also, these are some of the popular websites, resources for identification and information on plants of India. BSI eFlora of India. So that is a very helpful one, and also eFlora of India. This is the second one. That's it's a Google group, and there are more than 3,000 members. It's like a very active, online compendium of plants of Indian subcontinent. So there are more than 13,000 species already covered, and almost all species are illustrated. You can find multiple pictures. It's very helpful for identification, and it's easy to identify plant because there are experts and people interested in studying plants. They are in the team, and you can just post a picture and get the identification, and then you can go for authentication. So to verify, comparing with the herbarium specimen and checking the description whether it is matching or not. So to just get a preliminary temporary identification, you can use this. And there is Indian Biodiversity Information System, India Biodiversity Portal, Biodiversity of India, and FRLST, Indian Medicinal Plants Database. And each site will give you different information, and this medicinal plants database gives you all sort of information, vernacular names, and what system the plant is used, whether it is Ayurveda, Siddha, Unani, folk, like that. So there is no depth for information, but you should know which is authentic site and which is not authentic. This is a screenshot from the eFlora of India Google Group forum, and they are also maintaining a Google site for describing the species. So all species are illustrated. In the same page, you can compare different species belonging to a particular genus and representative pictures of different genera under family page. It's a very helpful one for identification. And another important tool for botanical authentication is herbarium, right? Without herbarium, there cannot be any botanical studies. Herbarium is the fundamental reference material for any kind of taxonomic research. So whenever you study a plant, the first thing will be, when you collect the plant, make a herbarium. Okay, that should be the standard. But not everyone is following, and that's why there are problems in the publication. So, and many people don't even understand the concept of herbarium. So, not the botanists, but there are other people, other scholars, researchers from other disciplines. They are also working with the botanicals. When the manuscript is ready and it is submitted to the journal, and they will come at the time, they will ask for herbarium number. So the reviewer is asking for herbarium number. Can I get a herbarium number? So it doesn't work that way. So when you start the study, when you plan the study, you prepare the herbarium and make sure it is identified correctly by a trained taxonomist, and it is submitted in a proper herbarium so that you can go back and verify whenever needed. So you know the process of conducting field studies, collecting herbarium, identifying the plant materials, using the local and regional floras. We used to conduct herbarium training courses for college students and teachers. So now it has become very easy because of virtual herbarium. There are so many herbaria they have digitized, including BSA herbarium and so many other herbaria. They have digitized the collections, and you can access, at least you can see the picture. Most of the time that will be enough to compare the leaves and the specimens of your plant with the herbarium, already collected and identified herbarium specimen. So it's very easy, very convenient. So you can access using a computer. There are so many other online virtual herbarium sources. You can get digitized type specimens, protolog. There is a biodiversity heritage library. You can get all sort of uh, literature. So these are some of the basics, and let's see how to authenticate botanicals. So these are some of the traditional methods, and these are the time tested, and even now these methods are considered very important. That's why even in pharmacopoeias they are including all these organoleptic macroscopic and microscopic methods in addition to other chemical molecular methods. Organoleptic method is one of the oldest methods. It used to be very popular during last century and all. So it is identifying botanicals based on their texture, color, and flavor, aroma, taste. So this method is efficient, especially in powdered materials. Like there are organoleptic experts. They can tell whether the botanical product is adulterated or pure just by observing these organoleptic characters. Like even in coffee industries, even now, only there are experts who can tell which coffee is the best quality, best tasting, and there is no machine to replace this technique, this talent. And macroscopy, as you know, this is the most common method of most popular method for identifying plants. All the floras, they use this method, microscoping method, for identifying the species. We don't need a hi-fi instrument, just a 10x lens is enough to study the characters. Only thing, we should know the basics of plant morphology. So I will not be going into details. You all know this. Like this is just an example how to use the macroscopic or exomorphic characters to identify species. Okay. So as you can see in this example, can anyone identify the number 1? This is Sida acuta because the leaves are acute, right? Number 2, Sida retusa because the leaves are retuse at apex. And this is Sida cardata, okay? And here is cardifolia, because of the cardate leaf. So just one character is enough to tell the species in this case. And here is rhombifolia, rhomboidea, okay? And this is spinosa because spines are present. So by observing the exomorphic characters, the morphological characters, the plants are identified. Generally, all the floras, there are regional, national, state level flora, they all use morphological characters to identify the species. But when it comes to the market, you won't find all the twigs with the leaves and flowers and fruits. So you will be finding only this harvested material, dried material. So how will you identify these? So that's why you need to have proper training in identifying the botanicals using macroscopic characters. I'm sure you all know this. It looks like black pepper, right? Actually, this material is mixed with the papaya seeds. So papaya seed is papaya seed is commonly adulterated to black pepper. So adulteration is everywhere. Okay, so almost, a lot of things are adulterated, whether it is milk, honey, water, rice, dal, everything is adulterated. But you should know how to distinguish. But fortunately, there are techniques to distinguish the correct from the adulterant. In this case, so how will you know if a papaya seed is mixed with black pepper? It's very simple. Take some alcohol, okay, not internally. Take some alcohol in a beaker and put the material that you think it's mixed with papaya seeds. So when you add the material into alcohol, the papaya seeds will float. All the black pepper corns will settle down. Okay, so this is one simple method. Similarly, there are some products you can just use water, but in this case, water will not work. So as I said, there are simple techniques available. But if you observe the morphological characters, how to distinguish the authentic from the adulterants, it's easy to authenticate. So these are some of the common adulterants for black pepper, like Embilla ribes. They say Embilla ribes, but Embilla xerium cotton is often adulterated to Embilla ribes, which is adulterated to black pepper. And also the 4 o'clock plant, the seeds look similar. So anything that looks similar, people tend to, it can be added and mixed up, either intentionally or unintentionally. Anybody can identify this? I'm sure you all know this is a star anise. Okay, but the problem is this is not one species. So in this sample, both Chinese and Japanese star anise samples are mixed up. So obviously they belong to the same genus, so they should look similar, right? But only the Chinese star anise is edible. That is used as medicine. It is also used for H1N1 and other viral disease, and it's a food and used as spice, very popular. But the Japanese star anise, Elysium anisatum, has a toxic compound anisatin. Okay, it's highly toxic, so it should not be consumed. It is not fit for human consumption. So, but sometimes maybe by mistake or maybe to increase the quantity, the Japanese star anise is adulterated to the Chinese star anise. So by observing the morphological characters, you can identify, or by microscopy, if you look at the surface of the star anise, so there will be prominent striations on Chinese star anise, but the striations are very not so prominent in Japanese star anise. This is the most expensive commodity, right? The saffron. It is also one of the most commonly adulterated product because it's not easy to collect the sample, right? That's why it is very expensive. It is collected from the stigmatic lobes of saffron flower, but it is very often adulterated with so many other things like safflower or tacitus, marigold, even corn silk is added. Here is the safflower, but the original authentic saffron should consist only the 3 stigmatic lobes. So the product will be considered adulterated even if it contains the stamens and the petal pods from the same flower. Okay, so the pure saffron should consist only the stigmatic lobes. So how will you identify the true cinnamon? If you go to the market, if you buy a cinnamon, how will you know that you are buying a true cinnamon? So there are different types of cinnamon, like cassia cinnamon, Chinese cinnamon, Vietnamese cinnamon. Again, there are different species of cinnamon are also used as cinnamon. But what you get in the market is most probably cassia cinnamon or Chinese cinnamon. Here is the one, the woody one. The first picture is the true cinnamon. It is also called Ceylon cinnamon or Cinnamomum verum. The old name is Cinnamomum zeylanicum. So in Cinnamomum verum, the true cinnamon, only the inner bark is used. When they collect the bark, when they harvest it, they peel and discard the outer layer, outer bark. So they use only the inner bark, which is thin, like papery, and when it is dried, so it coils multiple times. So that's why you see the multi-coil stick here. That this is the true cinnamon. The Chinese cinnamon or the cassia cinnamon, they don't discard the outer bark. So the whole bark is used, and because it is woody, thick, it doesn't coil multiple times. So it coils just once or twice. That's why it has a single or double coiled appearance. So this is one simple method to identify. And also because the outer bark is not removed in Chinese or cassia cinnamon, you can find the phloem fibers and also some of the lichen, because usually lichens grow on the bark in tropical evergreen forest, but in true cinnamon, it is not. You will not see these things. The true cinnamon is the best quality and also suitable for consumption. Chinese cinnamon is also edible, but it contains higher amounts of coumarin, which can cause health problems when it is consumed in large quantities. Okay, so we should know what plant we are using. So, of course, with the microscopy, we can easily identify. So when you see in the market, there are hundreds and hundreds of products. They contain wrong materials, incorrect ingredients. But the label can say one name, but actual ingredient can be different. Whether knowingly or unknowingly, they collect and include incorrect materials. So here is one example. As you can see, this product is available in major online stores. Anybody can buy it. People are buying and using it, I'm sure. But actually, they are using. Okay, here is the product label. So here you can see. So the product label says News Dela India. It means India nut. And also they say Alurates mollicana. It's right. Alurates mollicanus is called as India nut or candle nut. Which is edible, okay, but actually they are selling the seeds of Thevisia peruviana. You know, it's highly toxic. It contains glycosides, cardiac glycosides, which is highly toxic. So that's why the authentication becomes a priority. So the misidentified adulterated product can be lethal. So if this is taken in large, in higher doses, it can be lethal, right? It can be highly dangerous. That's why authentication is very, very important. So this is the plant candle nut. They're supposed to be selling, but actually they're selling Thevisia seeds. By using morphological characters, you can identify the species, but sometimes morphological characters can be confusing because sometimes similarities can be found. For example, different species of Tinospora can look similar, and they can look similar to even completely different species. That's why sometimes Pergularia is adulterated to Tinospora because they have the cardiform leaf. Anyone see here cannabis? When it comes to cannabis, everyone is botanist, right? Anybody can identify. But this is just to show there are so many different plants. They share the morphological characters. They all look somewhat similar, but there is no cannabis in this slide or in this slide. As you can see here, there are so many different species. They have the specific epithet that refers to cannabis, cannabis leaf, because they look similar to cannabis. That's why the species name says cannabifolia, cannabina, cannabifolium. So it means there are so many species that resemble cannabis. So misidentification is very, very common in the botanical market, and also many internet sites and the commerce. So let's see a couple of examples. So I'm sure you all know Embilla ripes, also called Vidanga or Voividang. It is a popular Ayurvedic medicinal plant. It is used for treating various ailments from indigestion to intestinal worms. It's a popular medicinal plant, but it is also a rare species. So whenever the species is rare, you can say that the product can be adulterated because, for example, Hoodia. Hoodia is a plant, very popular. It used to be one of the top selling botanicals in the USA because people thought it has um, appetite suppressant property. Okay, it has there are some studies, but there is no clinical studies proving the thing that it is used for weight loss. But if you see the market, there are hundreds of products, Hoodia products coming up because because of the popularity. But getting the product is getting the plant material is not easy because it is endemic to South Africa, where it grows in Kalahari Desert. It's not easy to just go and collect the plant. You have to get certificate of cultivation and you have to get the permission. It is also listed in the CITES, Appendix 2. So it's not easy to collect the plant. But if you see the market, there are hundreds of products. Hoodia products are coming up. So when we studied in our labs, we found more than 85% of the products are counterfeit. So many products did not contain Hoodia at all, although the label says Hoodia. And many products, our chemists found that there are no sufficient amount of Hoodia present in the product that will have some effect in our body. So more than 85% of the products are counterfeit. So when the plant is rare, you can say it can be adulterated, like yohimbe. So another plant, popular plant, very often adulterated. So similarly, Embilla ripes, which is present in Western Ghats, it is a rare species, but this is the one which is supposed to be used in Ayurvedic system. But there are 3-4 other plants like Embilla zerium and Misa indica, Myrcena africana. They are all substituted. Okay. When you see the internet, it's most of these. So here some of the pictures from the internet. I just searched. I did not go in search of wrong identification. I just searched for Embilla ripes, and these pictures came up. So all these pictures are incorrectly identified. Another grievous species. Even some popular websites like Bowl Systems here, they have listed all these pictures under the name Embilla ripes, but none of these belong to Embilla ripes, right? I think this is Kleinia, Embilla zerium. It's not at all clear here. So they say Embilla ripes, but they put Emblica officinalis, Philanthus emblica fruit. See, all these are misidentified pictures. Even the product, if you see, there are so many products of Vidanga. See here, whatever picture you see here, they are all Embilla zerium, not Embilla ripes. So all these are incorrectly identified. So we don't know what species they have actually included in the product, right? Although the label says Vidanga, sometimes they don't even mention the species name. And see, sometimes the powder looks green, black, brown. So it's very difficult to say what material they have included. That's why we need to be careful. And the product label should be accurate and should include the species name and mention which part is included. So here they say it's a Vidanga false pepper, Embilla ripes. Okay, this is from eBay. But actually they are selling black pepper, right? Yeah. they say false pepper. If this is false pepper, then which is true pepper? And this is a research article published in this journal. This is about Embellia ribes. The spelling is incorrect, and also this plant is not Embellia ribes. It is Embellia xerium. It appears so. So it looks like they have used this plant for the study, but the title says Embellia ribes. So what way it can be useful to the researchers? And there are many papers, many research articles. They have the way they have sourced the plant material and the way they have authenticated, it's highly doubtful if it is correctly identified. So in this paper, the authors have mentioned the dried seeds of Embellia ribes are collected from a locally situated medicinal practitioners and Ayurvedic outlets. So there is no information like how they actually identified, botanically identified, who authenticated, whether they collected any voucher material. So there is no detail. So you cannot say whether they have used Embellia ribes or Embellia xerium or Myrcena africana or Misa indica. Another paper here. So why I am saying this, whenever the students, if you are a student, whenever you write a botanical research article, make sure that you start with the right herbarium specimen, the right material, correctly identified. There are so many sources available to help you with identification, and there are so many floras and herbaria available. So in this paper, you see how they have written the author name. This is about Embellia ribes, pharmacognosy and phytochemical investigation. I'm sure you can tell what plant it is. So they have used black pepper instead of Embellia ribes. Yes, I'll show you comparative pictures. So this one is the black pepper. For comparison, I'm showing this. And the picture on the right side, this is the true Embellia ribes. So 100% this paper has used the wrong material. So when this kind of paper goes to the literature, it's kind of adding to the junk, So that's why you have to use the correct material, correctly identified. And this is another example, Phyllanthus. So when you say Bhuya Amla or Bhumi Amlaki, there are so many different species. Phyllanthus debilis, fraternis. If you see the commercial material, the material can have any of these species, either mixed or separately, because different people think different species as Bhumi Amlaki. So when the frontline stakeholders, so the people who actually collect the material, if they don't know how Phyllanthus amaris looks like, if they are given only the local name, so this is a problem because the same local name is applied to multiple species. And as you know, these are all different species. They are not same in their chemistry and in their properties, the medicinal properties. As you can see in this study, they have found Phyllanthus amaris is the only plant that has the phyllanthine and the hypophyllanthine, the active compounds in good amounts. If someone is using other species and if there is no effect, so they should not blame the system. They should not blame the Ayurveda thing is not working. So we have to make sure that the right material is used. So only then there will be a preferred and expected effect in our body, right? So just to tell you the differences, I'm sure you all botanists, you can distinguish, you can identify. But as you can see, Phyllanthus neruri does not occur in India. I have given the map. This is from the Plants of the World online. As you can see, these two species are not native to India, and they are native to Central and South America, both species. Although Phyllanthus amaris is very common in India, it is not native to India. It is naturalized. So all these purple colored distribution you can see. So because somehow people started calling the Indian plant as Phyllanthus neruri, although it is not neruri, but actual neruri occurs in Americas. So whatever plant we have is Phyllanthus amaris, and of course there are other species. There are so many research publications. They have published in the name Phyllanthus neruri. See, there are so many. So this is an analysis from Scopus. As you can see, hundreds of papers published from India, Malaysia, Indonesia. Phyllanthus neruri does not occur in these countries. So it means they have used either Phyllanthus amaris or some other species. So this gives a, incorrect information. It will have a chain effect, like the researchers who does the review article, they will combine all the papers published on in the name Phyllanthus neruri. So they will compile, and it will not give you the accurate results. And as I said, the misidentification is rampant. They just identify simply, like, I don't know if anyone verifies this or not. See, all these are misidentified pictures. So that's why you should be careful if you are comparing your plant with the internet sources. You got to be careful which site you are using. And all these are, they say, Phyllanthus neruri products. And all these from India. Again, the Bold Systems, this website. So they have included so many pictures under the name Phyllanthus amaris. None of them are correctly identified. I think this is Ziziphus, Phyllanthus simplex. It's not at all Phyllanthus. So far we have seen the importance of correct identification based on morphological characters, right? So when the botanical is in whole form, it's easy to identify morphologically. But most of the time, the products are sold as either powders or cut and sifted pieces, fractures. So how will you identify? How will you authenticate these kind of materials? So it is possible to authenticate even powdered material by using microscopy. So I will give you some example how we do by using microscopy. We use different types of microscopes for fluorescence, polarized light, and scanning electron microscope. So in this slide, you see the difference between the ordinary normal light microscope and the polarized light. So in polarized light, it's easy to view the crystals and starch grains because of the birefringent characteristic. So the top left picture, this is a Piper species. If you look at it under light microscope, it looks like this. But the same specimen, the same slide, if you look at in a polarized light microscope, you can see all these calcium oxalate sand crystals. Okay, this is an important character for identification. And in kratom, this is a Mitragyna speciosa. It's a popular plant, popular species. In the light microscope, the crystals are not so clearly visible, but you can see the Maltese cross or the birefringent characteristic. It's very clear in polarized light microscope. Also, Agathosma betulina, this is a characteristic feature of Agathosma. These are diosmin crystals. You can clearly see, and this is a key character for identification of Agathosma. This is, you can see the arrangement of prism crystals along the veins in Tinospora by polarized light microscope. And the fluorescence light microscope is also very helpful in identifying certain features like glandular glands and cell contents. So this is Eryngium vascular bundle. You can see the cell contents in the phloem and the xylem. So this is the Siberian ginseng, Eleutherococcus senticosus. The leaf section, if you see it under light microscope, the regular light microscope, you don't see the lipid, the oil droplets, but they are clearly visible in oil droplets. So using multiple type of microscopes will help characterizing different characteristics. Of course, scanning electron microscope will give you a high resolution, high magnification pictures. The top left, these are the E. coli bacteria found in a Tinospora commercial sample. Top right, this is a pit, bordered pit found in a fiber, trachea. This one is Salvia Divinorum. You can see the trichomes, glandular, non-glandular, and this is also Salvia Divinorum. You can see the arrangement of different types of glandular trichomes. So it's easy to see the detailed structures in a scanning electron microscope. These are colorized in Photoshop, just for clarity. Here is the original picture in black and white. So here in Salvia Divinorum, you can see when you zoom in, you can see the presence of calcium oxalate crystals within the cells. So when you study powdered materials, you should know what characters you are looking for, depending on the different part of the plant. So you'll be looking at different sets of characteristics. There are different types of trichomes. Even after grinding, you can find these characters in a powdered material. For example, this strigose type of trichomes present in Senna alexandrina. So if you see this type of trichome in Senna product, you can ascertain that Senna is present in the product. Similarly, the peltate trichome is present in olive leaves, and the stellate trichomes are present in Tiliaceae and Sterculiaceae members. T-shaped trichome is present in Vernonia cinerea. So these are very helpful in identifying the botanical, which is powdered. So again, different types of stomata, different types of crystals. So basically, there are 4 types of Raphette crystals published based on their morphotype. We recently published 2 additional types of Raphette crystals. I'll show you some pictures. So these are the Raphette crystals in cross section in scanning electron microscope. And here the top 3 pictures are light microscope. The figure C is the highest magnification, that is 1000 times, as seen in light microscope. But using SEM, you can view detailed characters. So when the morphology is confusing, microscopy plays a major role in identification. Okay, it's very supportive in determining the species. It is also used in the classification because some characters are species specific, like the stomata, presence of calcium oxalate crystals, and different types of trichomes, so on. So here we recently studied, compared the morphology and anatomy of 3 Tinospora species. Of course, all Tinospora, they have cardiform leaves. Tinospora sinensis you can easily distinguish based on the trichome, the tomentose character, but Tinospora cardifolia and crispa, it's difficult. Although the protuberances on stem, they are present in both species. So here are some pictures of Tinospora leaves. You can see the pearl glands, the unicellular glandular trichomes. You can see the shapes are different in Tinospora cardifolia and crispa. The picture with the trichomes, this is Tinospora sinensis. It's very easy to identify. But how will you distinguish cardifolia and crispa? If you see under scanning electron microscope, it's very easy to distinguish because Tinospora cardifolia has these kind of wax crystals, wax coating on the cuticle, leaf cuticle, okay? But in crispa, it is absent. See, it is smooth here, the lower left. But in Tinospora cardifolia, it is very prominently wax coated. But light microscope cannot help because the wax crystals will not be clearly visible. These are anatomy of the leaves and stems. These are starch grains, and also how the calcium oxalate crystals are distributed within the species is characteristic. And see different types of prismatic crystals, styloids were observed. In one of the previous slides, I mentioned about confusion with bay leaves because different species are called by the same name, bay leaf, and also used for the same purpose. But one of the species, this Umbellularia californica, is toxic. Some people are allergic to this plant. They can get severe headache just by smelling the leaf. So there are safety concerns. So that's why we need to identify the species correctly. So here you have Californian bay leaf, Indonesian bay leaf, West Indies bay leaf, Indian bay leaf, and the Mediterranean bay leaf. Laurus nobilis, as I said, is the true bay leaf. If the leaves are in whole form, it is easy to identify based on their morphological characters, like the venation, for example. In cinnamon, it's very unique. And also the texture and the aroma, organoleptic characters are helpful, but you can also identify based on microscopy. So how the oil cells and the mucilage cells are present and the number of palisade layer in the leaf, okay? All these characters are helpful in distinguishing the species. So here you see, we recently published this. You can see the sections of the oil cells. You can see the here mucilage cells, oil cells. This is the key to identify bay leaves. Microscopy helps in different ways. It can tell you the localization of alkaloids. For example, the alkaloids are present in the phloem here. So this is the leaf cross section. The pinkish colored cells are xylem. These are stained in fluoroglucinol and HCl. But these yellow substances are accumulated in phloem. So you can tell where the phenolic compounds are present in a particular plant. So which part has more abundant or less abundant chemicals. So by using different staining methods, you can tell whether it is a lignified element or phenolic compound or starch. So this is Phadogia agrestis. It's a popular medicinal plant from South Africa. There is a stem in the actual part is the part used is stem bark. So as you can see, all the compounds are accumulated only in the cortex and the phloem region. Okay, so here all these are phenolic compounds present only in the phloem region. So there are no compounds present in the xylem, the wood region, or the pith. So that's why only the bark is used in Phadogia. So there is no use of using the wood. So just only the peel, the bark of stem is used. So it is very helpful in localizing where the chemical compounds are present by studying microscopy. Of course, for cannabis, microscopy is one of the methods in addition to HPLC and other methods. So by microscopy, you can identify even powdered material or even the dried material you can identify based on the types and distribution of trichomes. So here you see three different types of glandular trichomes are present in cannabis and non-glandular trichomes. So here you see stalked glandular trichome. So the THC or tetrahydrocannabinol or cannabinoids, they are stored in the space, cuticular space of these glandular trichomes. So here the cuticle is broken, so you can see the secretory cells exposed. In the lower left picture, all these are secretory cells. And in the right side picture, you see the neck, which is broken to expose. There are 4 cells in the neck region. So another scanning electron microscope picture of the stalked glandular trichome. So these are the trichomes containing THC and CBD secreted. So this is the close-up of the secretory area of the cannabis trichome. So here you can see the polysaccharides and the secretions. These are sessile. So in the previous slides, we saw stalked glandular trichomes, and these are sessile glandular trichomes. And this one is the smallest, the bulbous glandular trichome. So you see the tiny one here, that is the bulbous glandular trichome. And in this picture, you also see the stalked glandular trichome. Okay, when you do microscopy, if you see something like this, don't mistake for glandular trichome. These are eggs of spider mites. So these are also found on cannabis. And also non-glandular trichomes. There are 2 types of non-glandular trichomes are present in cannabis. So these are mainly present in the lower part of leaf, I mean abaxial surface, and also present in the bract. So I'll show more pictures. So these are slender non-glandular trichomes. And these are the cystolith trichomes. You see the conical shaped short trichomes. When you take section, you can see the cystolith present inside the trichome. These are made of calcium carbonate. You can see in the polarized light microscope, the cystolith is clearly present. So by microscope, see more than 80% of cannabis, the illegal cannabis products are adulterated. So there are different types of adulterations happening and so much contamination problem you can identify by doing microscopy. They just add like so many different things, like in order to improve the flavor, the aroma, and also the appearance, they add body spray and the adulterated products called gritweed. Okay, you will find minute glass balls in the product. So these are just to increase the appearance, enhance the appearance, and also the price of cannabis. So that's why by microscopy you can easily tell whether the product is clean and authentic. And scanning electron microscopy can also help you in identifying contamination, heavy metal contamination. So you all know heavy metals are either added intentionally or unintentionally, or from the soil. It can be present in the plant. So you can identify these kind of materials, heavy metals, the presence of heavy metals, by using EDS, that is energy dispersive X-ray spectroscopy, or EDS, which is attached to scanning electron microscope. So these are some of the pictures of lead chromate. So this is the dye added to turmeric in order to improve the the color of turmeric, to make it brighter, bright yellow, so that the value will be more, they will get more price, but actually it is very toxic compound. It is not legal. They just add to, maybe unknowingly, or they just add to increase the value of the product, turmeric. So here you see turmeric powder, you see the starch grains of turmeric, and also the added chemical compound, lead chromate. So this spectra can tell you what are the minerals found in a plant material, in a product. So to summarize, authentication is a very, very important one. It can save lives, and it is very essential for research. So what we need is mainly training. So more people should be trained with plant identification, herbarium collection, the botanical basic botanical nomenclature, so that we can avoid. There are, as I said, there are so many methods, analytical methods, tests are available for authentication of botanicals, but only the physical method, the proper identification by using herbarium specimen and using correct nomenclature can prevent adulteration. So no other method can do this. Only the botanical authentication can prevent adulteration. So if you correctly identify the raw material using the proper methods by trained taxonomists, you can make sure that the plant material is not adulterated. So they use the right material. So it's like preventing a disease from happening. So this is very important. So this will save lives, and also the problems in the current problems in the dietary supplement industry. This is very important, training on plant identification and making herbarium voucher material, following GMP, GAP, GCP, all these good practices. Okay, and like certificate of analysis, most of the products when you buy, they will also provide certificate of analysis. In addition, there should be a certificate of identification. So whenever asked, the company should be able to provide a certificate of identification, how they identify the raw material, who identified, based on what characteristics, where the herbarium is submitted, whether the plant was collected from the wild or cultivation. So all this information will help authenticate the plant material, right? And every step we have to follow the good manufacturing, good collection practices, good harvesting practices, good agriculture practices. Okay, so this way the identification and authentication of botanicals will be very easy. So the moral of the story: macroscopy and microscopy methods are reliable and effective in identifying and authenticating botanicals and detecting adulterants and contaminations and substitutes, and also to check to ensure the quality of the botanical. And thank you very much. I thank Dr. Khan FDA and NSF for their support. Thank you all. If you have any questions, yeah, thank you.
Yeah. Thank you so much, sir. It's a very detailed and informative session on macroscopy, microscopy for authentication and quality control of botanicals. And some of our participants are from Ayurveda, Unani, and homeopathy colleges, sir. They sent a very good feedback, and some of students, some of participants would like to interact with you, sir. Karthik, Mr. Karthik.
Sir.
Yeah.
Hello, Karthik.
Hi, sir.
Yeah. Yeah.
Hi.
Can you take our participants?
Yeah, sure, sir. Who raised the hand should I take into the panel?
Yes. Otherwise, ask them to talk from there. No problem.
Okay, sir. One. Yeah, 7777 code whoever it may be, you can talk now. And Sri Lakshmi Garu, you can talk now. You can unmute your mic. Sri Lakshmi Garu. Sujana Garu. Rajchandran Garu, you can unmute your mic and you can ask the questions or you can interact with the sir.
They can also contact me by email.
Or
so Karthik, here is
Karthik, some of them posted in chat box. Just read out so that sir can.
Hello.
Someone is asking, sir.
Hello.
Yeah, please tell me.
Good morning, sir.
Good morning.
Sir, very informative presentation regarding authentication and control of botanicals. Sir, I want to ask that you said that cinnamon, the stem that is used is been adulterated with cassia also. So how? Could you explain it out?
No, cinnamon cassia is also sold as cinnamon. If you see the market, there are cinnamon material coming from different parts of China, Vietnam. So they are different species. They are from different species. But the true cinnamon is the Ceylon cinnamon, which is Cinnamomum verum, also called Cinnamomum zeylanicum in the past.
Okay, now my question is, sir, that cassia bears bitter compounds.
Yeah, it has more higher amounts of coumarin.
Coumarin. Okay.
Yeah.
Based on that, they have utilized.
Yeah.
Thank you very much, sir.
Thank you.
Sir, this is Sri Lakshmi.
Hi.
Sir, can you hear me, sir?
Yes, ma'am. You can ask.
Yes.
Yeah. Good morning, sir. Vijay, sir, it was really a great presentation and thank you very much. And sir, I would like to ask you, like the cinnamon, the original variety, which is mostly available in Sri Lanka and other regions. But here in the supermarkets, local supermarkets and all, mostly we'll be getting the cassia cinnamon, that is Chinese cinnamon. So what are the ill effects you think using this cinnamon? Because it is very tough to find the original cinnamon available in the. Of course, it may be available, but most of the time it is only the Chinese cinnamon which is sold. So what are the ill effects of prolonged use of this cinnamon?
See, even cinnamon, the cassia cinnamon, can also be used as long as it's used in small amounts. So if you are taking multiple, many spoons, teaspoons per day, it's not good. So if you can get the true
publications, are they available to read, sir? Because I'm very much interested to see what all tags you have used in the anatomical studies, the microscopy studies. I mean, those beautiful pictures you have captured. I would like to know the methodology. Sir, are the research articles available to read, sir, or can you please share?
Of course, yeah. You can check my ResearchGate site.
Sir.
Just search by my name, you can get, and I have shared most of my articles, the full articles, or you can just contact me. I will send you the
Yes.
papers.
Thank you, sir. Thank you so much, sir.
Thank you very much.
Yes.
If anyone is having any queries, please raise your hand.
Yeah.
Sir,
can I ask a question? Hello.
Yes, sir. Yes, sir. Raichandran, sir. Yes, sir.
Yeah. Hi, Vijay Shankar.
Hello,
Great
sir.
to hear you.
Vanakkam, sir. How are you?
Very nice. It was excellent presentation.
Wow,
Fact,
thank you so much.
Yeah, we are proud of you. Like I did follow your publications very interestingly.
Wow.
Most of them are quite interesting and fascinating research you are able to do. And in fact, now you are trained in taxonomy and you are now excelling in anatomy and authentication of botanicals. Very interesting to know you and see your presentations, Vijay Shankar.
Wow.
Excellent.
Thank you so much. It's really great.
I wanted to contact you, but
me
other
too.
commitments. Yeah. Fantastic. I thank the organizers for bringing in Vijay Shankar here to this forum. Actually, people like Vijay Shankar should be utilized maximally in Indian context, because there are a lot of adulterations, though in spite of molecular studies and other things, people are still unable to authenticate the botanical specimens and raw material. So you are an insight to many of the youngsters, Vijay Shankar. Great work.
Thank you so much. All your training, and you are my guru, right?
I just shared the slides to Dr. Ravi Kumar also. He was happy to know about this. Great.
All your training. Thank you so much.
Okay.
I hope you're all good.
Yeah, we'll talk later.
Okay, thank you.
Thank you, sir. Thank you so much. Geeta ma'am, you can talk now. Unmute your mic and you can ask any question. Sir, some of them posted in chat box. I'll ask some questions.
Okay.
Which variety of Phyllanthus bhumi amalaki is available in Karnataka?
There are different species available in Karnataka. Phyllanthus urinaria, Phyllanthus amaris, but not Phyllanthus niruri linnaeus. So see here, the author name also is very important. You can see if you are referring to a local flora which used the name Phyllanthus niruri, if you are referring to that particular reference, you have to quote Phyllanthus niruri ocnon linnaeus. Okay. The actual Phyllanthus niruri occurs in Americas. It does not occur in India. So that's why the correct, the author citation is very important. So whatever is treated in Indian floras, it is Phyllanthus niruri ocnon linnaeus or Phyllanthus amaris.
Yes, sir. One more question. Someone is which variety? Sorry, sir. Can you please name some sites which can give correct plant identification?
I have already shown multiple sites, right? There is POWO, Plant of the World Online. Okay, Plant of the World Online .org, I think. You can check the plant list, but you should check the check multiple sources to find the most latest name, the accepted name. And you can also check the Medicinal Plants Name Services, MPNS. That is one of the most, recently updated databases. And there are so many other sites also.
Yes, sir. One more question, sir. What's your opinion on DNA barcoding in identification of adult fish?
That's a good question. DNA barcoding is a very helpful tool in identifying speech. Okay, but it has to be like any other method. It has to be used um, for the right material. Okay, there has, you know, for instance, you cannot use macroscopic characters for identifying extracts and powdered material. Similarly, DNA studies cannot be used poor quality sample, very old sample, and extracted material, right? Because the DNA quality will not be good. So that's why we call it as a fit for purpose. So you have to use the right method for the right material, and always use multiple methods to get the more accurate data. For instance, a microscopy can tell, you can find whether it is a leaf or a flower or a root or stem by doing microscopy, but DNA cannot distinguish the different parts. So the DNA is same for all different parts. So for instance, in Ebrus precatorius, the leaves are edible, but the seeds are toxic. So you should know which part is included in a product, which part you are consuming, right? So the part is important, and that's why you need to use the correct method and always use multiple methods. It's a good question.
Yeah, one more question, sir. How to differentiate root or stem of Rubia cardifolia, both macro and microscopic?
I have not studied the Rubia cardifolia, but it's not very difficult, I guess, by identifying morphologically. I'm sure there are microscopic studies available for Rubia cardifolia.
Yes, sir. One more thing. The anatomy and intracellular substances varied according to the environment. If so, then how can we conclude the correct species?
That's a good point. So we cannot just identify based on cellular components, the cell inclusions or chemical compounds. So in anatomy, we study the architecture of the plant, the different types of tissues, and also presence or absence of calcium oxalate and other diagnostic characters. I said you can identify, you can localize where the chemical compounds are mainly present in a particular sample. So it cannot be, you cannot identify species based on this character. By doing histochemical tests, you can identify which type of compounds are present, like phenolics, lipids, like that.
Yes, sir. One final question.
Yeah, to study the chemicals, you have to use chemical methods like HPTLC, HPLC, UPLC. There are many methods available.
Okay, sir. One of the participant is asking about the career options, sir. Sir, any workshops in your future in your lab, or can we attend them through DBT or DST, or any other funding available to attend? Please let us know.
Can you repeat the question, please?
Sir, one second. Any workshops in near future in your lab, and can we attend them through DBT or DST, or any other funding available to attend? Please let us know.
We conduct training programs on different aspects of dietary supplements. There are different stakeholders from the dietary supplement industry. They come and attend the training courses. And we also conduct training on cannabis authentication. We include multiple methods, macro, microscopy, different types of chemical methods, and also DNA barcoding, everything. So it will be a package training in different techniques. So far, we have conducted only on-campus sessions. Off-campus, I'm not sure. Maybe you can send me an email, so I will give you update. So when is the next program? You can also search for NPTEL website. natural products training laboratory within University of Mississippi. You will get all the information.
Thank you, sir. And final one more question, sir. Sorry, sir. How to know about the purity of the finished products we get in the market like leaf, churna, and so on?
For that, there are different methods like NMR and HPTLC, HPLC. For finished product, you cannot use macro and microscopy methods, obviously. That's what I said. You have to use the right method for right type of material, right? The fit for purpose test. You can use any chemical methods for finished products to quantify and also to identify.
Thank you, sir. Thank you so much for your wonderful presentation, and almost very very interesting to see every presentation and colorful also. And never like there is no bore or anything when we
of course, plants are not only beautiful externally; they are also beautiful internally. You know, the anatomy, it's very beautiful actually. So anybody will love the subject.
Because when we see the presentation or anything, it's very interesting to know about what it is exactly. Because some people who don't know also, interested researcher when we see about the pictures or anything, very good presentation, sir. Very beautiful actually. Thank you, sir.
Thank you so much, and thanks, Dr. Vijay Bhaskar Reddy, for the invitation, and for Dr. Pullaya, and congratulations for organizing the important seminar on plant taxonomy. We need more such programs. Sir, thank you so much.
Thank you, sir. Our co-organizer, Dr. Nagesh Rao, would like to share a few things with you.
Yeah, sure. Okay.
Over to Dr. Nagesh Rao.
Yeah, thank you, Vijay Shankar sir. Thank you for your very detailed, very highly informative talk. Definitely, I hope our participants, especially from the medical background, homeopathy background, will get the lot of benefit from you. We expect that your cooperation in future also. Our students or our scholars will need your support and guidance. We welcome your support also in the days to come. Now I request Dr. Mahmood Ahmad from the Manu, Assistant Professor of Botany, to propose a vote of thanks to the present lecture.
Thank you, sir. Thank you so much.
Thank you.
Is it audible, sir?
Yes, sir. It's audible.
Sir, please turn on your video also, please.
Yeah. Yeah, I think it is visible.
Yeah.
On behalf of the organizers, I am proposing vote of thanks of this first session. First of all, before proposing vote of thanks, let me congratulate entire team of Nizam College for organizing this excellent workshop of 5 days. And I should congratulate Dr. Vijay Bhaskar Reddy and his entire team for this bringing in a very good manner. I should even say thanks to Professor Vijay Bhaskar Reddy, and I would like to inform you all that Vijay Bhaskar Reddy is not is known to me since a long time because he has worked on the flora of university campus, our university campus. Many of the people don't know that one of the central university is located in Hyderabad, just adjacent to Central University of Hyderabad. Another central university is located in Gachibowli, that is Maulana Azad National University. And here we have very good campus. In our campus, even the flora work has been done by Dr. Vijay Bhaskar Reddy. I should say that he is one of the good taxonomists in Hyderabad. And today lecture presented by Dr. Vijay Shankar Raman on authentication and quality control of botanicals, a macro and microscopic approach, was really a wonderful way he has presented. Initially, he has explained how the researchers are making mistakes in terms of naming of plants, their origin, and even in authenticity. So we should be very careful before publishing. We should be very careful. And further, he has even explained about the use of fluorescence light, scanning microscope, how the fluorescent light microscope helpful to see the internal structure of a plant. As just now he said that plants are, one of our colleague was saying, beautiful pictures you have shown. So in turn, he has replied that plants are not only beautiful from external side, internally they are very beautiful, because it is really, because whenever he was showing the pictures, all the in detail with the glandular structure, even with the cell content in xylem and phloem, really it was awesome. The picture he has collected from the microscopic technique are fantastic. Each and every photograph is very, very clear. So at one side, I would like to say that we are not familiar with these techniques. So we should have some training. We should have certain type of trainings, like this micro macro technique training. Even if Nizam College or the Usmania University is organizing in future course of time, I'll be very grateful. And finally, in one single sentence, by going through all his complete presentation, in nutshell, I would like to say in one sentence about our resource person, uh, all the elements, are so mixed in him that nature may stand up and say, here is our Dr. Vijay Shankar, here is our Vijay Shankar. So thank you very much, sir. Thanks a lot for being with us for today's session. And also, I thank organizers, especially our Vijay Bhaskar Reddy, as well as Nageshwara Amanchi. I should thank Dr. Nageshwara Amanchi, because even when I registered, I was not sent any communication. On the last day, when I approached Dr. Nageshwara Amanchi, on the same day, on the same time, he has sent a message and he has incorporated my name. So I am very grateful to Dr. Nageshwara Amanchi and Dr. Vijay Bhaskar Reddy, and finally Dr. Vijay Shankar Raman sir for his today's excellent lecture. Thank you very much, sir.
Thank you, sir. Thank you so much. And
thank you, sir. Thanks a lot. Thank you so much for nice words. Really appreciate. And it's a very good idea, like training and microscopy. Yeah. I think we should. It's needed. It's very important. I'm glad you mentioned it. Oh. We conduct training programs also on microscopy. So it can be. You can check online in our University of Mississippi website. Maybe we can provide. I don't know, online training. So, but students can always contact me with any questions, whether it is taxonomy, macro, microscopy, anytime. Thank you all. Thanks, Dr. Vijay Bhaskar Reddy, Dr. Sachikala ma'am, and Dr. Nageshwara for organizing such a wonderful and very important training course. Workshop.
Thank you, sir.
Thank you all. Thank you very much.
Thank you.
Thank you, sir.
Thank you, sir.
Now I request Dr. Shashikala ma'am to introduce next speaker.
Sir, I am very sorry. I forgot to mention the name of Dr. Shashikala ma'am. Yeah. I'm very sorry.
Never mind, sir. Never mind, sir. Never mind, sir.
I think I met sometime in Nizam. I met even Nageshwara, sir. But
yeah.
Very sorry.
No, never mind, sir. Never mind, sir.
And thank you, sir, Makbul sir, for a great vote of thanks. Thank you. Yeah, next speaker, Dr. Karupaswamy is ready. And Madam Dr. Shashikala, madam.
Shashikala, ma'am.
And no
yes,
problem.
Sir. Yes, sir.
Yeah, please, shall we go with
Yes, sir. Good morning, all the
Good morning to
participants.
all. I think no need introduction. Already I have.
Yeah. So just it is a formality. Already we had your class yesterday, 2 days back. I think you need no introduction. The introduction is for the unknown people, but you are already known to all the participants with your excellent lecture and with your inspiring content in your lecture. So without wasting much of your time, valuable time, because you are well known to with your expertise, you are well known to all the participants and also organizers. So I'll invite you for the talk, sir. Thank you very much, sir. Thank you. Thank you, sir. You can
Thank you,
start the
ma'am.
session, sir.
Yeah, thank
You
you
can
very
start
much.
the session. Thank you.
Yeah, I'm trying to my. You can share my slides. Can you share, please?
Sir, please share your screen, sir.
Yeah, this some problem is there. Already I have sent my PPT to Surjan.
Sir, I'll share, sir. I'll share, sir.
Okay, please.
Yes.
Yeah, meantime we can start. So very good morning to all the participants and organizers of the webinar on plant taxonomy. I'm very happy to meet you once again to all this in this forum. And today I'm going to speak about molecular taxonomy. You know, you have enjoyed since the first lecture, Dr. Sanjay Prasad and Professor Pullaiah and all others. You know, previous lecture was it's excellent and mind-blowing on that authentication of plant identification by Dr. Vijay Shankar. So it is very interesting also. So now I'm talking about molecular taxonomy. You know, I'm not going into very detail and techniques of molecular taxonomy. Just I want to give a outline of molecular taxonomy because some other sessions also there. The organizer arranged for some techniques in molecular techniques, molecular taxonomy. So I'm giving only the outline of molecular taxonomy. So last few days you are hearing about the taxonomy, but you know taxonomy is the science that explores, describes, names and classifies all organisms. Now, as you know, all botanists or biologists, these are the common principle. So this science that provides baseline data for all fields of biology and plays a vital role for society, but it is also an independent, complex and sound hypothesis driven scientific discipline. You know, plant taxonomy is the one of the ancient scientific discipline that emerged thousands of years ago, even before the important contributions of Greeks and Romans. So in 15th and 16th century, the plant taxonomy benefited from the great navigations, the invention of printing machines, the creation of botanic gardens, and the preservation method of herbarium techniques, etc. So in parallel with the development of morpho-anatomical data,
see the previous lecture, it was developed by the invention of microscope only. That much was happened. Nowadays we are doing authentication and everything. So the parallel, the development of morpho-anatomical and subsequently emergence of plant classification, artificial, natural, with permanent naming systems for plants. Finally, we reached the evolutionary thought. So finally, very recently, the cladistic theory and rapid advances in DNA technologies allowed to infer phylogenies and to propose true natural genealogy-based classifications. So now we are going to the molecular taxonomy. What is molecular taxonomy? You know, every organism has several composed of several molecules. So in this taxonomy, the classification of organisms on the basis of distribution and composition of chemical substances in them. So this is very simply the molecular taxonomy is defined. So then how it is defined means that it is because of techniques. So molecular techniques in the field of biology have helped and established to genetic relationship between the members of different taxonomic categories. So this is very clearly defined molecular taxonomy. Molecules are there. We are going to relate the organisms, or we are going to infer the organism because basis on the molecular relationship between them. So this is the molecular taxonomy. Next one. You know, it was started from the origin of species. So before that, also I told that taxonomy is ancient science, but the people, when they were looking into the molecule only after the this man has published Origin of Species by Charles Darwin. So this is giving more insight into the biological changes in the biological world. You know, before that classification was different. After the post-Darwinian classification is entirely different. And before that, the biological approach was entirely different. So after publication of this Origin of Species, every biologist, they have looked into the seriously, and they have changed all the systems of their approach for biology is very different. So he has given more insight about the phylogeny. So he is the basis of the evolutionary system or biological, that phylogenetic system. Next one. No, he was drawn, he drew a lot of the diagrams like that. It's called phylogenetic tree. Now it is called as phylogenetic tree. But without the basic knowledge at that time, in the 1830s, he drawn a beautiful diagram, phylogenetic diagram in his own notebook. That's a diary. He has, just to look into it, now only we drawn. So this is comparable to the modern phylogenetic tree. The Darwin's phylogenetic tree, just based on his assumption, he has drawn a very beautiful phylogeny and what is the root, what are the branches, what are the in between the gaps in the branches, that is nodes and internodes, what it is depicting. Excellently he has explained in his notebook. But it is a original, I think, phylogenetic thought. These are the phylogenetic thought. Based on this only, now we are drawing the modern phylogenetic tree. So next. No, another one important thing we have to look into it. So this man, everybody knows, all biologists must remember his name, Ernst Haeckel. So he has written a book on trees of life in 1866. See the year, 1866. He has drawn a beautiful that phylogenetic tree of life in his book. You see that left corner, you can see that. So this phylogenetic tree he was drawn in his book in 1866. You can see that every tree has very broad basal portion. And when it is in the branches and other things, it is going into smaller, and again it is divided into smaller, and canopy is having much smaller portion. So this is the original phylogenetic tree. He has given that another one important thing, we have to remember him. He has given a lot of words about to biology, ecology. He coined the term ecology. And another, he coined the term only phylum and phylogeny, protista. These are all the words coined by himself. And he has drawn beautiful phylogenetic tree. But after 150 years, what will happen? See the next slide. Yeah, this tree of life now we have drawn like this. You can see the root portion where it is derived from this root portion. Even for the protista, archaea, eukarya, bacteria, there is no any root. It's a rootless tree. So like that, after 150 years, we gained a more knowledge. We invented more things. But even though now we are getting the phylogenetic tree is like that, all the trees are rootless. That means our the ancient, the first life is clueless. Where from we are coming? Where from we are coming? Which is a root stock for the particular life? We are not having any knowledge because of already it was extinct. All the primitive world life or primitive flora and primitive fauna, they may be extinct. We could not derive from that the genetic material or genetic makeup of the or genetic idea of the primitive life. So that's why the until available organisms or available plants, only we are doing like that, all the DNA and analysis and everything and drawn trees like that. So that's why we are getting the rootless tree. So this is the idea of phylogeny. Phylogeny means where from we are coming. We have to find out our ancient. So the ancient we could not found out means the tree is incomplete. So that's why nowadays we are getting the phylogenetic relationship means only available taxa. So we are we could not have a idea to fill the gap in between them. So the relationship of the one taxa to another taxa only because of available DNA or available genetic material, but in between some gaps are there. So every time we are telling the man is evolved from monkey, but monkey to man, a lot of gaps are there. The time is different. Man was originated different time and monkey was originated different time. In between a lot of organism was there. So that was missing. That genetic material was missing. That's why we could not have could not relate with the other one organism to other organism directly. So this is the problems in when drawing the genetic relationship. Next slide, please. Why introduce molecular systematics? Okay, now we are looking into only on taxonomy, plant taxonomy. Why we are introducing this type of the systematic or molecular taxonomy, molecular systematic like that? You gain a basic understanding of tools available, what they can or cannot offer and how they work. We should know first why we are looking into the science, the molecular science. What are the importance of this? You must think about it, or this is only the tool for taxonomy. This is alone is not for a taxonomy. Molecular is only the not for taxonomy. Now, previously it was given the very beautiful lecture on authentic identification through microscopy. That was a tool. Anatomy is also not the tool. So morphology is another tool, basic tool of taxonomy. So molecule also one of the tool for taxonomy. Second thing is to provide you with the vocabulary and concept used by the molecular systematics. So it is giving the very limited that vocabularies are there. We should understand the concept and vocabularies what we are using in this molecular systematics, and not to teach you how to go into the lab and start doing the work. So this is only based on experiment. So molecular systematics, so only based on experiment, and it is one day, within a day you can learn the technique. So within a day, you can learn the technique. Suppose if you go to the molecular lab, and what are the system, what is the PCR system, how to handle the extraction of DNA, what are the protocol, you can do. You can learn it from a single day, but basic taxonomy is not like that. Taxonomy, you may not, you continuously learn, mean also you can't complete the whole portion of taxonomy. It is a very broad science and very broad sense also there. And molecular taxonomy is the wave of the present and future. Maybe it is the present time we are rely on this subject, and maybe future also we hope a lot of that opportunities are there in this molecular systematics. Next one. Yeah, this is a molecular, again it is what are the molecular phylogenetics. So molecules are there, different molecules are there, proteins or carbohydrates, the macromolecules are different kind of macromolecules are there in organism, but which molecule we are going to look in for phylogenetic system. The molecular phylogenetic is a study of evolutionary relationship among biological entities. You must understand this one. Biological, because lot of creatures are there in the world, but we have to relate with one taxa, one organism to other with the phylogeny or evolutionary relationship based on some molecules. So it may be the individual, or maybe the population, or maybe the species or higher taxa, but we can relate with all these things based on some molecules in them. So by using combination of molecular data, either it may be a DNA or RNA or protein or any other metabolites, because plant system they have a lot of metabolites. So based on this, we can relate in this system. So this is the molecular systematic or molecular phylogenetic. Next slide, please. Yeah, there are a lot of landmarks or milestones in the developing of this subject. So whenever we are studying any subject, we should know the history. History is important. The first 1953, they elucidated the structure of double helix DNA by Watson and Crick. Everybody knows. So after that, 1972 is the development of recombinant DNA technology first time. The isolation of DNA and other things, it was they discovered or invented the technology because a lot of machine was invented at the time. And 1977, the first complete DNA genome to be sequenced from the bacteriophage. And 1977, Alan Maxam and Gilbert published DNA, partial DNA sequencing. And 1984, Medical Research Council, they have published the complete DNA sequence. And 1987, they have the machine was, the automated DNA sequencing machine was invented. 1990, and they have sequenced many number of bacterial genome, Mycoplasma, Escherichia coli, and Saccharomyces. Like that, the prokaryotic genome was sequenced. And finally, you can see that in the year 2001, the draft sequence of human genome is published. So the development, the very fastly developed this kind of molecular science since from the 1950s to now the 2000s. The last 50 years, the development of this DNA technologies was tremendous. So they have input a lot of the new concept proposed in the biology based on the DNA, because DNA is the life molecule. Next slide, please. So after that, we have every day now the new technology has come and introducing some kind of new things for the biological world. So problem with morphological data. So why we are already, I told you that, so we have to go for this kind of technology means some kind of disadvantages in the previous system. So since the taxonomy was originated, we are using morphological data, but now we are relying on molecular means. What are the reason? So convergence and parallelism in morphological characters, reduction and character loss. Many number of species, the evolution may not go in all the organs. Already the evolutionary thoughts are different, either progressive or retrogressive. So evolution may not happen in all the organs, or maybe in the single organ through the progenies like that. So these are the characters. Morphological character has a lot of difference in different species or within the same species also. So phenotypic versus genotypic differences. So same species, it may have produced the white flower or maybe colored flower within the same species, some small genetic differences. So evolution of homology, misinterpretation of change of polarity, limitation of number of characters, phenotypic plasticity. The single tree, when it is growing, means you can see the different types of leaves. That's called plasticity. So these are the reason for we cannot rely on morphology alone. So now objectives of molecular taxonomy. What are the objectives? Reconstruct the correct genealogical ties among biological entities. So we have to relate the organism in the correct sequence of the phylogeny. where from originated, which is our ancestor, what are the intermediates. So like that, we have to identify or find out or relates the correct sequence of phylogeny or sequence of origin. So that is the main objectives of molecular phylogeny. Estimate the time of divergence between biological entities. This is most important, which time it was originated, because our rate was originated the crores and crores of years back. But after that, slowly the evolution was happened, organic evolution was happened as per Darwin's thought, and slowly the number of organisms will be increased, and then which time it has originated. So now everybody knows the Jurassic period, it was there, our earth surface dominates the gymnosperms. So now it is completely dominates the angiosperms. So when it was started, the angiosperm phylogeny. So like that, we have to estimate the time of divergence between the one plant group to another plant group, or one organism to another organism. This is the main objective of molecular systematic. And third one is chronicle the sequence of events along evolutionary lineage. We have to correlate all the other data, either the climate or from the internal changes in the organism. So we have to correlate all the thing for the evolutionary lineage of particular groups. So this is the main objectives of the molecular taxonomy. Next slide, please. So what are the gene fragments we are used? Already know molecular taxonomy is entirely depending on either DNA or RNA, because that is the life molecules. Already we know that. So what are the fragments? Which portion of the DNA or which portion of the RNA is giving information, more informative in because we have different groups of organisms in the world. So first one is prokaryotes. Because most of the organism, we have certain common portion of the DNA, because we are all coming from the same lineage. Maybe the first life only it is giving the all other life based on the cell theory. But like that, prokaryotes, it is having RNA genes used for species identification. You know, most of the prokaryotes, RNA is very stable than the DNA. So RNA genes are used for species identification in prokaryotes. Similarly, animals, birds, and fishes, small region of the mitochondrial gene, that is cytochrome oxidase gene, is highly informative. For plants, RBCL gene sequence and MAC gene sequence are informative. RBCL means it's important gene or protein for the synthesis of enzyme ribulose 5-phosphate carboxylase. You must know that it is an important enzyme for photosynthesis. It is only the fixing the carbon dioxide, the first step of photosynthesis. The enzyme is involved to fix the carbon dioxide. So all the green plants, it must have that protein or that enzyme. So the gene must be there in all plants. That's why it is a particular gene we have selected. Similarly, in mitochondria, maturase gene sequence, it is found uniformly in almost all the plants, even sometime in animal also. But in animal system, mitochondrial, the cytochrome oxidase gene is important. So another fungi is somewhat odd man out from the plant, because non-chlorophyllous, it is having the chromosomal DNA region that is called ITS region is informative. Next. Molecular markers, then markers. Markers means which is important one. So we have to have a standard, the portion of the DNA as a marker. So that is a molecular marker used for comparison of all the organisms. Then only we can get the idea of the variation between the organism. Then we can relate to all the things. The molecular markers can be characterized as type 1 marker and type 2 markers. So type 1 markers are associated with genes of knowing function. Type 2 markers are associated with genes of unknown function. These are the basic difference between markers. So, and another marker is called allozyme marker. So, allozyme is a different kind of the marker type. It is based on the type 1 marker, but even though they encode the associated with some functions. So, this is called allozyme marker. And another marker is microsatellites. Recently, the many number of taxonomist or this phylogeneticist used for microsatellite marker, and other neutral markers also there in now it is in market. Next slide, please. Yeah, this for plants. Now we are only for plants. All other organisms are also there, but many number of phylogenetists now they are working on different groups. We are concerning only about the higher plants. So it has many many number of genes are highly informative. So they are nearly 16 gene portions are highly informative in flowering plants. So among those, I have given ATPF to ATP pH spacer. These are the MATK gene, RPCL gene, RPOB gene, RPOCL gene, like that. These are the genes are highly informative. Nearly 16 genes are highly informative, the genes are gene markers in plants. The assessment of recoverability, sequence equality, and the levels of species discrimination, they recommended the two locus combination RBCL METKG as the plant pargod. So now everybody is aiming to the pargoding technology. So for simple identification or authentic identification, we are thinking about pargoding technology is much useful, and like that these two genes are working in the technology is highly useful, particularly RBCL and MATK, and other genes are also there, but we should find out. And now the system is working like that, and CBOL and Consortium of Barcoding of Life, they have rely only on these two genes for higher plants, only RBCL and MATK. So next one is pargod region of plant. Already I told you that you can see that plant cell is having different cell organelles. Even every biologist, that mitochondria and chloroplasts are semi-autonomous organ. They have their own DNA itself. But these are all highly informative. You can see I have listed out all the informative gene in higher plants. Particularly they sometimes found in chloroplast, or sometimes found in the nuclear genome, or sometimes found in the mitochondrial genome, like that. Because nuclear DNA is the conserved region are called ITS, internal transcript spacer. So that is a conserved sequence. So like that, mitochondria and chloroplast also there. So mitochondria and chloroplast DNA is very comparable to the primitive organism like bacteria. It is a circular DNA. They have a limited number of the base pairs of DNA. So some of the regions are conserved. These are all highly informative for our phylogenetic studies. Next slide, please. This is the chloroplast genome of Vitis vinifera. You can see the number of genes are there. The totally 1060 odd base pairs are in the DNA. And you can see some of the regions, large single copy regions, repeated regions, and small single copy regions, inverted repeated regions. So like that, but some of the genes are also listed out. You can see that Rubisco subunit, that ribulose biphosphate carboxylase subunit is located very specific site of the chloroplast DNA. And like that ATP synthase, this is also important enzyme for all kind of reaction that in biological reaction, ATP synthase, this is located very specific region. So these are used as some marker genes in the system, plant system, particularly the chloroplast genome. Next slide, please. Yeah, some chloroplast coding genes and their function. Already told you that these are having a very specific function. Each and every genome and genes are having very specific function. The ATPB genes, largest single copy region in chloroplast, they're located in a very specific place. Their function is the subunit of ATP synthetase. It functions in the synthesis of ATP via proton translocation. So every enzyme, their function is very specific at the site of the biological system. So all these things, RBCL, it is only the Rubisco, it is fixing the carbon dioxide. Similarly, MATK, maturase K genes, which function is in splicing of second introns from the RNA transcript, and NDHF, small single copy region of chloroplast, the subunit of chloroplast NDDH, dehydrogenase. So this is important function. All the genes are important function. That's why all organism, it must be there. So we have to select the common uniform genes. It must be present in the region. So very conserved region. That's why we are selecting these genes for study of phylogenetics. Next slide, please. Yeah, now it is ITS region. This is an internal transcript region. It's from nuclear DNA or nuclear DNA. You have two sites, ITS1 and ITS2 is common for all the plants. Is common for all the plants, and also the length of the gene only vary from plants to plants. But this region conserved site is common, and the length of the gene only vary from plant to plants. So this is only helping to compare the relationship of the taxa or plants or any groupings of plants based on the amount of the sequence present in ITS1 and ITS2. So this ITS region, the nuclear DNA. Next one. Yeah, the same thing. You can see I have compared some of the ITS region from different, is a bacteria. Bacteria also there, the first row you can see this. This is the region ITS1, ITS2. It is the blue color portion. You can see there the length is varied. The number of base pair is varied for that constituting the ITS region. So second one is plant. You can see in between the DNA regions. So some the base pair, other base pair are also different. So these are helping to the identification. Next one is animal and chloroplast DNA. The plants and animals, you can see the after splicing, you can see how it will be arranged. So these regions are informative. We can infer the phylogenetic relationship based on the number of base pair in one organism and other organism. Just we are comparing. It is much close, mean it is closely related. If it is highly differed, the number of gene in one organism to another organism, that is fair from the evolutionary link. This is a kind of comparison of the phylogenetic system. Next one, please. Yeah, here also you can see there it is a mitochondrial, sorry, it's a chloroplast DNA. It's a somewhat large number of genes are there. Here also I marked the ITS1 and ITS2 regions. Next, please. Next. Yeah, here now it is a technology. So previous one. Now it is a technology. So very simple. Now the PCR technology is very popularized. First, we should know the isolation of DNA. So what kind of technology used for isolation of DNA? It is varied for plants and animals and other microorganisms also. So because of this technology developed, now the protocol is standard, but when we are doing our laboratory or your laboratory, some variation will come because it is entirely depending on what are the chemicals used for the isolation of the DNA. So which portion of DNA we are going to use, that is also what they depending on the enzymes we are going to splice the DNA. So this technology is very, so that's why all the molecular technology is not showing the uniform result in all the laboratories. It will come the differential result from the laboratory to laboratory because it is only working with a chemical system, but natural materials are similar. So here just we are isolating, but most popularly we are using CTAB isolation, DNA isolation for our molecular systematic, but now a lot of molecular other techniques also it has come for isolation of DNA. But even now the plant system most we are rely on herbarium specimen. So if once it will fix the alcohol or any other chemical, the extraction of DNA is very difficult. But even though we have to overcome that things, now we are extracting DNA from herbarium material also. But the fresh material is very easily, and we can extract the DNA very easily from the all other kind of techniques also. But no, CTAP method is commonly used for the extraction of plant DNA or isolation of plant DNA, and then PCR machine. It is just a machine. It is having, explain you how it is PCR will work, and then application, sequencing, and next analysis and comparison, and finally we can compare the taxon. So this is now it is helping to another step for barcoding, DNA barcoding. Next slide, please. Yeah, now everything is behind the molecular technology is behind the 4 letters, set GT or ASG, AC like that. The 4 letters, the how many number of genes we are getting from the informative region. So that is only comparable to the taxa to taxa, and finally you can see the comparison sequence like that. So what are the similar sequence? It is arranging the homologous region, means the organisms are closely related like that. So everything is based on the 4 letters, 4-letter codes. Next slide, please. Yeah, now the technique PCR, it is everybody knows today polymerase chain reaction because of the enzyme polymerase. That polymerase we are using for the multiplication of DNA, and it means it is process used to amplify DNA replication into thousands of copies within a minute. With how many cycle we are using means that much the multiplication of DNA fragment will get it from the technology, and it is work only based on DNA, with the purified DNA and heated denature and renature, denaturation and renaturation of DNA material. Next, just to click it. Yeah, it is also this technology is depending on primer used, either short or conserved DNA region complemented to the ends of DNA to be amplified, that is called primer. So already we should have a standard primer, then only we can pick up the right portion of the DNA from the organism, and it will be multiplied or by means of the enzyme DNA polymerase. So that machine is called the PCR machine, or the PCR reaction. Next one. Yeah, this is a stepwise technology. This is a commonly knows all biologists today. The solution heated, just denature of the DNA. It's a cycle of denaturation and renaturation. That is a basic concept of our PCR reaction. So just a double standard DNA, it will become single standard. Then we are changing the temperature because of presence of enzyme, all raw material, it will constitute our region. Again, it will synthesize another copy of the a complementary copy of the sequence. So again, it will be go to the temperature changes, denaturation, renaturation. It will be repeatedly occur. We'll get the multiple copies of our required part of the fragment of the DNA. So this is only the basic or very simple PCR technique. Nowadays, all the laboratories we are using today, but it's also but highly sensitive. If we want to prepare, or if you want to get it the very particular region fragment of the DNA, we should work very carefully on this technology. We'll get the right sequence of the particular region. So if you have any contamination in DNA, or if it is not extracted properly, or if you are not getting using the proper enzyme or proper marker enzyme, you will never get it the right portion of the enzyme. So our complete result will not be reliable. That is not a correct one also. These are the things we should know before going to do the technology and our techniques. We should thoroughly learn all the protocol and the working system, then only we can go with the molecular technology. Next one, please. Yeah, next. So after DNA sample, we will get it, means we'll comparison, just we'll compare the things. Next slide. Yeah, here the next one is electrophoresis, and this PCR is always associated with the electrophoretic technology. Then we can separate all the fragmented DNA. We need to have only a particular portion, and our portion we have a marker already, and standard marker is also there. And electrophoretic technique, we can separate the right fragment what we required. That fragment we isolate it and then go for the sequencing. These are all automated nowadays by machines only. So this electrophoresis, just we can. Now we have a lot of mechanisms. So after that we can go for sequencing. Yeah, next one. Next one, now it's available, a lot of molecular methods available. Every day we are introducing a new kind of DNA extraction mechanism and the sequencing mechanism and the using different markers. Every day we are introducing new things. So I have listed only the limited number, only 15 types of the biosystematic molecular methods available, but still it will go. A lot of another recent technologies also available, but basically we are using for molecular systematic only about this 15 techniques. One is RFLP, everybody knows, restricted fragment length polymorphism, and the next one RAPD, random amplified polymorphic DNA, and AFLP, amplified fragment length polymorphism. These three are so very primary the molecular technology, and prime technology also we are using commonly for all kind of molecular techniques. And another things, variable number of tandem repeats, VNTRs, this also used. PCR based several marker based that methods are also available. Multiple arbitrary amplicon profiling, like these are all, every technology, it has its own merits and demerits. Every one, it is particularly for it is work with some specific groups. For example, algae means we cannot go for all this technology. Any one technology is only suitable for algae. Bryophyte means it will suitable for one particular technology. That's why these many number of technologies are available for working with the molecular methods. But all the technology, but a single technology alone cannot used for all organisms. We can have a very specific technology, specific group is working with a particular type of molecular methods. Next one. Now, advantage of molecular data. What are the advantages? So already told you the morphology are having several problems. So molecular biologists, they are telling the morphology is having several problems for identification. Either sibiling species are there that morphologically very similar. Two species are there morphologically very similar. We call it a sibiling species. So like that, these are the problem for identification and the classification, even in taxonomy. So here are some advantages also there for molecular data. So one is molecular entities are strictly heritable. So because DNA and RNA are the heritable material, so it is strictly heritable. The description of molecular characters is unambiguous. Unambiguous means without doubt we can have a very sure for that character. So that's why molecular characters we are selecting. There is some regularity to the evolution of molecular traits. So gene only expressed and it is giving the traits of all organism. So that's why that it is regulated. It will not change. We are thinking about it is the gene is permanent one, genetic characters are permanent one. So that's why we are selecting. Molecular data are amenable to quantitative treatments. So this also, but sometimes it will be varied. But homology assignment is easier than with morphological traits. Molecular data are robust to evolutionary distance. We can get many number of gene sample or many number of gene markers in a single system. It is getting a very robust for studying evolutionary distance. Molecular data are abundant. So every organism, it must be there. The genes are genetic material. So it may be abundant in all the organism. Less time consuming. For morphological data means gathering of morphological data will take more time, but we can get it very simply from computer, and we can after the sequencing all the data assembling is very simple, and time consuming process is very less. So these are the advantages of molecular data. Next one. Yeah, now so our own examples also already know that these are the group everybody knows. Carloma. In Indian Carloma, already the previous lecture also I told you that the Carloma now is segregated into different subgenera, but now it is previously it was called Carloma. We have published a book also, monograph of Indian Carloma, Carloma sensulato. So you can see the flowers, all are Carloma, but vegetatively you can see all are succulent, succulent cylindrical or quadrangular leafless stem. It is a vegetative character, but you can see the flowers. The flower characters are entirely different from species to species. One species is coming axillary single flower, and another species are coming apical umbellate flower, like that. So these are the difference between the species, a lot of difference between species to species, but even though we can identify with morphology is also simple, but apply the molecular systematics is very sure for identification of this particular group. Next slide. Yeah, it is a common protocol for extraction of DNA, PCR that extraction of DNA, PCR amplification and sequence. And these are the common procedure we also have done for this, the particular group of Caralluma in India. Next slide. Yeah, now we must know that the sequencing is not that much simple for gene. Whenever you are studying molecular systematic or try to sequence, try to extract the gene, you cannot have the complete sequence of gene. Or if you try, means very difficult to get the complete sequence of gene. So partial sequence of genes only nowadays we are doing. All the gene, whatever may be the submitted, these genes only, their sequence are only from the partial genes, not complete genes. The first complete genes, it was sequenced from this plant Arabidopsis thaliana. It's everybody knows it is a experimental plant, the permanent experimental plant for all kind of our molecular biology today. It's a tiny plant. Every biologist know that why they have selected that plant, and it is easily grow in experimental laboratory, and because very small number of chromosome, and every chromosome has a number of genes, and location of genes also very specific. So characters are the highly defined in this particular plant. So that's why they have the first sequenced plant, the complete sequence of genome from the Arabidopsis thaliana for plants. So now many number of plants, corn, paddy, corn, banana, many number of plants now they have sequenced completely. But even though this is the plant, the first sequenced completely. Next one. Yeah, this is a DNA alignment. After we are getting that code, so after sequencing machine, it will give the 4 letters, sequence of the 4 letters, and next we are grouping into the all 4 letters for analyzing or inferring the relationship. So taxon 1, 2, like that 8 taxons are there, and they have arranged all the sequence, all the DNA alignment we have, and character coding in the last. You can see that number of sites, how many number of sites are very similar. That is a coding. If it is more close, the taxon will be more, relationship is more close. If it is highly relates or distance are there in one taxa to other taxa, one sequence to other sequence, that we can arrange in the very spatial, and it is relates to this is a more close or more distance. So this is only just characters coding with the sequence. We can align the after alignment of the DNA sequence, we can get it the inference by means of the character coding. Next slide, please. Yeah, this is another alignment DNA coding. So today these are all doing computer itself. So we are not, manual coding is not possible. Everything in the computer, you can get it, the colored things, how it is related to one taxa to another or one sequence to another. So automatically it will give the similarity and differences. Morphology also identification is based on similarity and differences. In molecular systematic also, it's similarity and differences. How much similar to one sequence to another, or how much distance from one to another. So this is only the comparison of taxa, or comparison of one to another by only because of the similarity and differences. So all this alignment, it will show the similarity and differences. So based on this, we can infer how much it will relates to one sequence to other. So this is only coding. And after this coding, we will go for phylogenetic analysis. That is also based on computer. Next one. Yeah, it's a weighing data. Actually, it will not show the computer, but it is automatic. It's a program. The weighing the DNA of sequence only by the program. It is called matrix. it is a statistical program actually. So we can weigh 0 means it is no difference. So here you can see that this taxa A or species A and species A, there is no any difference because its own DNA, it is highly similar. And another next one, you can see that it's a very close numbers. Just one single barcode is very, single region is highly restricted region is very close means. So like that we can relate all the taxa, how many number of taxa analyzed or how many sequence we got it, everything it has been compared, and will get inference from the sequence of weighing, the weighing technology. It is only by the computer program, not by manually doing anything in this molecular technology. Next slide, please. Yeah, now it is the computer will give inference of the weighing that statistical information. It will give this table. Now ITS means, so this gene we have taken, number of taxa we have taken, the total number of how many characters we have drawn for this, but number of the region, and the parsimony informative, this is statistical informative for the phylogeny. This many number of statistical information we got it from for this per 39 taxa, and what model we have used in the particular analysis. So like that all the gene. So now how many number of gene we have studied for the single organism or many organism, all those things pooled into the computer data, it will show the similarity and differences. So this analysis, parsimonious analysis. In computer, we can say it is called the parsimonious analysis. You will get the information, the relationship or the distance of the taxa, how much. So one taxa to another, we are getting derived from the computer information. Next one. Yeah, this is also same. Sometimes when you are sequencing, some genes are missing. Some genes will be missed in the sequence. So what that is also we have to do manually align that genes. That is more important. That's why sometimes this technology, phylogenetic technology or sequencing may not be true, because at the time of sequencing or at the time of some the isolation protocol or any protocol in the PCR, it will be missed some base pairs. So manually we can align. It is only based on assumption. There also we can have some statistical techniques for the alignment of gene, which gene is missing or which code is missing in particular sequence. Sometimes it will be missed. So that is based on only assumption or statistical method. Only we are filling the gaps of the sequence by manually. So this is somewhat I know that negative inference we are getting from the molecular data. Sometimes the full genes we cannot sequence. Already I told you that full gene we cannot sequence, but in a partial genome also some of the codes will be missed, or the missing region or gap will be filled by ourselves or by means of computer with some statistical technique, only based on probability. It may be there, it must be there, or it based on assumption we can fill that code. Next slide, please. Yeah, this is whenever if you are doing that molecular technology, already many number of genes it was deposited in the gene bank or some database. So permanent world database banks are there. They have deposited a number of sequence. You can get it from the sequence, or you can extract, you can get a fragment, you can get that amplified fragment or sequence, you can compare it. So we are also getting from, I told you, all this Carlumas species and Bauschrosia, Carlumas, we are collected from different region. So these are ITL and TRNL, TRNLF. These are the genes are submitted into the gen bank, and we have done this, extracted the gene and submitted for gen bank for analysis. Next slide, please. Yeah, these are also same thing. Next. Yeah, now it is, you can see the phylogenetic tree. So this is a phylogenetic tree. It is analyzed by the computer program, and I will tell you what are all the advantages and disadvantages for this analysis also. Now for this Carluma species, Indian Carluma species, we got it the maximum likelihood tree based on ITS sequence. It's only based on the the nuclear DNA. You can see there, and these are all the other Carluma species or other related species also there. This information we are getting, or genes we are getting from the GenBank or other databases. So only these, the box you are showing green color and the magenta color, these are only we are our own study. You can see the Bauserosia and the topmost the clade, you can see the Bauserosia, and in between it is Carluma. There are two portions which has come. Previously all are called Carluma. All are called Carluma, but now we have clearly aligned in the two different portions. The Bauserosia is above, in between some African elements are coming, and also Carluma clade is separated, based on ITS sequence. So ITS, internal transcript sequence, it is clearly aligned two different groups. Indian Carluma is segregated into two different groups, Bauserosia and Carluma. In between, some African species also coming. You can see the Bauserosia, Freeria indica is coming in the basal ancestor for Bauserosia. So now yesterday, that the previous lecture, I told you that now Freeria indica is transferred to Bauserosia. Bauserosia freeri now is a valid name. So it is more close to Bauserosia. Maybe it is an ancestor for Bauserosia, because Freeria is leafy plant and that Bauserosia is a leaflet plant. Yeah, next one. Yeah, this is another chloroplast DNA regions for the same group. Here also you can see that Bauserosia is arranged in a separate clade, and other Carluma is having into two groups. So Indian Carluma clearly segregated into the two groups based on chloroplast DNA also. You can get it, we can get inference from this phylogenetic tree, and all other African genera and related species, it is arranged into a separate clade. Next one. So combined also we can analysis. Next one. Yeah, here combined the tree for both the ITS sequence and the chloroplast DNA sequence. So you can see that it is more closely related with the Bauserosia and Carluma. So now Carluma alone also we can see there, here Bauserosia clade, here it from branched from Bauserosia clade. The Carluma clade, here it will branched from the Carluma clade. So these are all the important that based on the genes, tRNA and tRNA tree, you can see that clearly segregate the branches of the group, and this indicates some of the points. So where the nodes here, the junctions, the junction is the origin point. If we properly analyzed with the some programs, computer programs, we can get a time of origin also. Next slide, please. Yeah, here also same thing, Bauserosia clade. You can see some numbers on the clade. So these are also the statistical coefficient, how much it is related. If it is 100%, you can see here 100% means these are all originated from the same point, but more or less similar. These are all more or less similar species, Carluma poindriana, Carluma pauciflora, Carluma crenulata, at the same place. Another one thing, the phytogeography also important. So this implies the not only for molecular information, and also it reflects the other characters. Even nowadays it support morphological character, phytogeographical distribution, and even endemism, and all other characters, even anatomical characters, everything we can compare and give the support of this molecular. But molecule only it is not giving full information. So we have to compare everything. So these clades are also important. How much percentage of difference or percentage of similarity between the one group to another groups also indicate. It is by means of statistics we can put this number also. Next please. Yeah, here also same. So likelihood based trees. So now there are two separate genetic lineages of Carluma sensulato in India. One is based on ITS and other sequence. One is, you can see morphology support. Here example for morphology. So one group is a Carluma group. So this side is a Carluma group. It's the attenuate erect stem. It is having axillary flower. So floral, petals are like that. It is rotate or linear like that. Here you can see that the umbellate inflorescence, it will be on the tip of the succulent stem. So these are our group Baucerosea. The two lineages separated. One is Caralluma, another one is Baucerosea, and other some minor characters also there. Pollinia, pendulous, Pollinia erect, and you can see the petals and everything. So these, how can be calculated these things, or how we can infer all the information only based on the gene means, but because of phylogenetic tree. So it was introduced by Henning and everything, everybody knows. So here indicates some of the important things. One is scale. This scale also put based on the statistical tool of analysis in the computer. So this is a distance. When we are analyzing any phylogenetic tree, we should carefully look into it for the distance of the this particular portion or branches. So species A means, topmost species A, you can see the distance. So it is when it has originated, and the time is giving the length of the branch. So distance between, and also vertical line, it is distance between the two species. Here you can see species A and species B. This distance, the vertical distance, it is giving the distance between the two species, how it is close, how it is how much amount of relationship between the two species, it is giving the vertical line. And also the branch length, it is giving the also the distance of origin. And node, this node, only the originate point, only the particular time it has originated from the two species from their rootstock. So this indicates the time and evolutionary time and evolutionary period, how much it was there. And even evolution is not there means it stopped the evolution, or still it is going into the evolution. That also it will indicate the phylogenetic tree. Suppose if we have analyzed with this good marker, or it is suitable for the particular group of taxa with analyzed with the good marker. Next slide, please. Next slide. Yeah, next please. Oh, some problem is there anyway. So here, like that, we can infer a lot of things from the molecular techniques. It's okay. So here, the ultimate thing is we can get an inference from the relationship of taxa, one group to another groups, or what group we are going to analyze means we can get inference from the molecular data. And so I want to tell about the yeah, it has come, I think. The steps in phylogenetic, I already told you the different steps for the one is extraction, then sequencing, and then analysis part by means of the statistical methods or tools we can analysis. So it has given in a slide steps in phylogenetic analysis: assemblage and alignment of data set. Different genes we have already got it from our experiment, and it could be analyzed from different other that data site also, database site, NCBI, and these are the programs: BLAST, ClusterLab, MAFT, and TCopy. These are the different programs for alignment of the sequence, and we have to align all the data is to be from this by these methods. Next, building phylogenetic trees. There are also these programs are some of the programs are freely available on computer or internet. POP, PML, and Philip. These are the program for construction of phylogenetic trees. And these are the another statistical method to analyze evolutionary distance among clades. So already told you the branch length and the vertical portion of the length, where from the branches originated, nodes and internodes, everything it has been analyzed by means of that matrix method. And finally, they produce rooted trees by molecular clock. So it has been, we should have selected the very proper rootstock, or the method, we can get it we can get it a right tree, that is a rooted tree. So otherwise it will, every program it will give thousands and thousands of phylogenetic trees. So we are getting all the publication, it will show only one tree, but we will analyze the program, we will get it thousands and thousands of tree for a single running or a single data set. And we have to select which tree is suitable for our own description. That has to, we have to select, that is also by the statistical method. So maximum parsimony algorithm applied for tree estimation. So now we have to estimate the tree because 1,000 trees are coming for our data set. So which tree is suitable to explain or suitable for our group, we have to select, and for applying based on the computer program. So you can see for a single tree, so these are all based on only 5 species. You can see A, B, C, D, E, 5 species, all are for same data set. The data set is same, but you can get a different types of trees. So this is a kind of tree, this is a kind of tree, these are also kind of tree. So every trees are having same, but which one is suitable for our interpretation, we have to select. That is by means of the computer program. You can see some of the three rooted tree. So here also root is there, second one, the second row also rooted tree, but you can see the last row there is no root. Where from it is originated, we can't infer from that, because unrooted tree, it's our data set also it will come, but we have to select the very proper, which one is a property, and also time, the scale we have to put mean time of origin, and the species alignment. You can see the second row probably it is suitable for the interpretation, but the first one, second, the last row we cannot interpret because that is not properly aligned. So any program, it will come a lot of trees, and we should select from we pick up the very right trees, we got it. So how we can select means, these are the methods are there. The nodes, each represent, the node is represent a taxonomic unit. So wherever the nodes are there, that is represent a taxonomic unit. Branches definitely the relationship between the taxonomic unit in terms of descent and ancestry. So where from these branches are, it is indicating the branches of ancestry, where from originated, that's root stocks are indicating. And next one is branch length. Already I told you, usually represents the number of changes that have been occurred in the branch. So this branch is only the limited length, or it is going to extend like this, means whatever the changes are there in the particular portion, that can be indicated in the branch length. If it is short length, means the very short time it is changing, one that gene is changing into another, it's diverting to the divergence is taking place in the very limited time. If it is a lengthy branch, it means there are a lot of time it is taking further, that much changes of the particular gene, or increasing the number of gene, or for divergence divergent of the evolution. So like that, here branch length is indicating the relationship of the taxa. So like that, lot of examples. So here I already told you, unrooted tree, we can't infer the ancient or rootstock. We could not, the tree will not help to identify the ancestry. So like that, these are all applied only by means of computer program. It is not manually, we cannot do anything. So we will get only the inference from the phylogenities, and interpretation only ourselves. So manually or artificially we are interpreting the getting the data from the computer itself. Next slide, please. Next. Yeah, some examples are there. Lupha tuberosa. Even now also POWO, that's the Plants of World Online, the synonym of the doubtful species they have given, name is given, but you can see the bottom line synonym of that within a blank, they put the blank. This piece also on confusing species in Cucurbitaceae, Momordiga cymbalaria previously, and Lupha tuberosa, Trichosanthus tuberosa. Trichosanthus is a genus, snake god. Lupha, everybody knows, that is also a vegetable. And Momordiga, that is a bitter gourd. But which belongs? This plant is confusable for on taxa for taxonomist. Either it is a snake card, or it is a that sponge, that plant, loofah or momodica. In Telugu, it is called Kakrakaya or Kasrakaya, usually found in some Kurnool, Kadapa, Anandapur, that district in Andhra Pradesh. In Tamil Nadu, we have only few districts, Madurai, Sivagangai, Ramanathapuram, only few districts are there. It's endemic to South India. But until now, the taxonomic problem was not solved. And we have done some taxonomic the molecular systematics of this plant. It is goes to Loofah tuberosa, it's more close to Loofah tuberosa, and we have given the suggestion to change the name into Momodica cymbalaria to Loofah tuberosa also. But even now it is synonym of just they have put blanc, and because the more confusion was there on this particular species, but it is a good vegetable. And another, we can, through the molecular technology, we can find some of the new species also. So we have published one species, Hydrocartyl collimaliensis, in collaborated with Professor A.K. Pandey. Previously, he was the professor in Delhi University. Now he's, I think, Vice Chancellor of Manasarovar University. And we have done the phylogeny of Hydrocartyl in India. We found that one species collected from Tamil Nadu, one population, it is entirely different from all other Hydrocartyl, and we have published a new species based on the molecular technique, Hydrocartyl collimaliensis. We can also have, because molecular technology can help to give the information or variation or from the correct identification of the taxa through the molecular technology also. Next slide, please. Yeah, this is angiosperm phylogeny. Now, I think yesterday you might heard the lecture of Professor D. Narasimhan. He was given very wonder lecture of the APG fourth classification. A lot of confusion, and still it is there a lot of confusion. Still many families are not correlated properly with the settled to the classification properly in APG4 also. You know, you must think about the amborales, nymphaeals, ostrobales, chloranthales. You know, we are saying this is a pro-angiosperms. But what are the ancient or rootstock for these four? It is more confusing because we are not getting the proper taxa. So it may be extinct through the periods. So that's why we have related only available taxa is like that, the pro-angiosperm, and after that magnolids. In between monocots are coming. So all our previous classification, it has been monocot and dicot. The flowering plants means we have easily segregated from monocot and dicot, but now it is monocots are it is coming in between dicots, two dicots here, basal dicots and the pro-angiosperms. In between it is coming the monocot. So probably this could be derived from the time of origin may be the same for monocots and dicots. So this is only we can get it. Monocot already it was there in the Bessay system. Bessay, he was wonderful, the founder of the phylogeny, and he has inferred the monocots and dicots was originated at the same time. And even Ranalian school also they have thought the monocots also in the same time it was originated at the olden time. But now we have classification like that. Yes. You got it, that material also, I think, yesterday, as everybody knows how we could derive the basal angiosperm or phylogeny. Phylogeny is what could be inferred from the phylogeny or phylogenetic classification. So these are all excellently explained in this material, but even though you should have that separate eye on the phylogeny or classification or modern trends. So how we can approach this classification? Either it will be come to the practical usage, how we can be used. So these are the question will be arised. So what are the other step we are going to the molecular taxonomy? Is we'll go for next slide. Next slide. Yeah, here next, after this phylogeny, now we are going for global gene barcodes. Barcoding is only the ultimate the identification technology. But now we no need to study that basic information or basic taxonomy, or we no need to the principle, we no need to study after coming this molecular systematic. So ultimately it will go for the par coding, I will tell you later. And what are the problems in molecular systematic? Now, you can see the redundancy of genetic code, convergence not seen, selection not at DNA level. What we are doing now, our molecular technology only based on DNA or RNA. You know, the what molecules or what genome, what technique, what analysis for you, this is only depicting our result. So our result is rely on the first what molecule we have selected, what genome we have selected, what technique we have selected, what analysis we have followed. A lot of diversity was there in this all these techniques. These are only based on the technique. Suppose if you have selected a wrong molecule, either DNA or RNA, so that your result will be changed. Or wrong technique, you know, I listed several techniques are there for isolation technique or that analysis technique. If you are selecting wrong technique also, our result will be changed. So the previous one, I think, you know, how many of you know the only one amino acid is for one code? I have given that genetic code, the table of genetic code. So you should look into it. All other amino acid, among 24 amino acid, only one amino acid is having a single code. All other amino acids are having multiple codes. Multiple codes. Only that is a methionine. Just you look into it. I have put in a box. So methionine is only the amino acid is having single codon, and all other amino acids are Y. And another you have to think about it, which amino acid is abundant in the world, abundant among the biological organism is a methionine. Methionine is having single codon, but it is abundant in the biological organisms. But other organisms are having more than 2 or more than 3 or more than 4, but they are very less. So why? So these are genetic redundancy. One gene is not for one amino acid. It may be, it is giving some other amino acid also. So this is a problem for the identification or sequencing. Another is gene trees. I already told you this is only by means of our computer program or based on the very specific and selective program. So it is also a lot of problem when we are selecting the trees, either compare orthologous gene copies, not paralogous gene copies, or big uses in molecular systematic all organism, but get information from 2 genes. Next slide, please. Here another problem is gene sampling is too little. How can you say that only the evolution within the RBL gene, or only the evolution in the MATK gene, or only the evolution in the cytochrome oxidase gene for animal? No. Every organism is having a lot of genes. Even human genome, we have a 100, even only the functional genes are 5%. Remaining 95% of genes, redundant genes or junk genes, we do not know the function. So how can you select only a too little sample, only few genes? How can get you information for all this phylogeny or arranging a complete classification for a whole kingdom of plant? So this is one of the disadvantage when we are studying molecular systematics. And taxo sampling too little, placeholder issues. And another things, just we are selecting and extracting from only a single leaf. Single leaf, you can get a whole genome for a whole complete information from that particular single mitochondria or particular portion. No, we have to collect multiple samples and check with all the multiple samples are having the same gene codes or similarity within the isolated fragment. We have to check it, but we are not doing like that. We are molecular because technology is highly expensive. And isolated texts are long branch attraction. Another one thing, suppose monotypic genera is there, that will not included in our, even we cannot correlate with the taxa. Even now several families are there in APG 4th classification also, that is not properly settled, even though they have segregated. Like that Bentham Hooker, they have at the time of classification Ordinus anomaly, on order they have created Ordinus anomalous families. They are all even unrelated families are completely put in one order, Ordinus anomaly, order anomaly. They're like that. So here also order anomaly is there. Even after the molecular world, now we are doing many things in molecular. Now also the order anomaly is there. We are not getting any information from the families for put in a right place. And all have issues in basal angiosperm phylogenetic. Already I told you though, basal angiosperms, now we are fixing our pro angiosperm, basal angiosperm, like we have fixed in the APG fourth classification. Even now a lot of debate is going on for it, is it right or not. Next slide please. Yeah, no, nothing in biology makes sense except in the light of evolution. The famous quotation was defined by Theodius Prabazhansky. You can see there, previously every man, after the publication of Origin of Species or the Darwin's theory, everybody is looking into the evolution. This is based on only assumption. They have given the evidence based on ecology, genetics, taxonomy, cytology, development, morphology, biogeography, everything. It is probably the backbone of evolution. Evolution may create or change, or it is giving all those diversity of subjects, all those diversity only because of evolution. So they have clearly explained. But now what we are putting, next slide. Now in the center of, previously it was that evolution. Now you can see there, now we are artificially just we are incorporated into the place of evolution. We put phylogeny. The phylogeny is completely artificial thing, but evolution is a natural phenomena. So now phylogeny we have given in the center portion. We are all looking into the phylogeny. We are all getting support to the ecology, genetics, taxonomy, cytology, development, morphology, biogeography, everything we are giving the evidence. But phylogeny is artificial things. So now we are, is it a question mark? Nothing in evolution makes sense except in light of the molecular systematics. This is a question mark. Now we are putting a lot of artificial data. So now the next step we are going for the DNA barcode, because this small DNA fragment, it is giving information for to develop another technique that's called barcoding for the identification. So DNA barcode is short genetic sequence that can be used for biological species identification. The first Herbert, he was proposed that concept in 2003. So named after black stripes used to identify products in the supermarket. You know, universally now they are identifying all those things only because of that the dark and light lines only. So one of the potential plant DNA barcode is coding RBCL. Now we are thinking about, so because of that global consortium of biological, global barcoding of life, consortium of barcoding of life, the working group, they have identified. So in plant system, RBCL is a good informative gene for the developing barcode. Next slide, please. Next slide, please. Yeah, this is the man. Paul Herbert is the founder or the father of barcoding. I think DNA barcoding is a technique which is species identification performed by using DNA sequences from a small fragment of the genome, either mitochondria or chloroplast. Mitochondria for animal system, chloroplast for plant system. A small fragment system only enough for creating our barcode, and it can be used for the identification system. Next slide, please. Yeah, this is significance and limitations, because every techniques or any technology, there are the two sides are there, like coins. There is advantages and disadvantages. Here some significant limitations are there for the technology. Either phenotypic plasticity and genetic variability in the characters employed for species recognition can lead to incorrect identification. Even molecular identification also sometimes it will not be correct. If you are developing barcode also, that may not be sense the correct things, because the plants and nature has created and changed even time to time, and that is always evolved, and it is right to set in the environment. So that's why it is also when time will go, means this technology will automatically it is abolished. We cannot go through and overlook morphologically cryptic taxa which are common in many groups. You know, I told you that as a sibyling species are there. That time we can use, but even also it will reflect. Sometimes the character means it is the same character is there in the another species also, it will give the false result. The morphological keys are often effective only for particular life stage or gender. Many individuals cannot be identified. Although modern interactive versions represent a major advance, the use of keys often demands such a high level of expertise that misdiagnoses are common. So it may also give some false diagnostic methods because the only limitations are there. Only thing, the successful DNA barcode means short enough to be quickly sequenced. We can sequence quickly and easily identified in all species of organism, variable enough to provide a unique sequence for each species. Size of the sequence, it may be a very smaller, say 600 to 700 base pair, and universality. So these are the some of the limitations and for using the successful DNA barcode. Next slide, please. Yeah, now everything, now it is a plant is there, the DNA is there already, but we can get it. But there are two schools are there, and two types of people are there in everywhere. Now advocates, they are all advocating this recent techniques, are accepting the recent development. They are telling ID all species, but opponent won't work. We can identify the based on molecular technology, we can identify all the species they are telling, but another orthodox group, it may not. So previous one, please. Yeah, these are the things important. So there are two groups are there. Even though advocates are telling discover new species, but traditionally destroy the traditional systematic. So orthodox people, they are telling you are killing the taxonomy, systematics or traditional taxonomy, like that. The speed of the IDs advocate the new technologies. Maybe it can be the time-consuming process. You can identify very immediately, even without knowing the basic principle. But the opposites are they are telling the service industry, right? That revitalize biological collections, but they are telling your collections are pseudo taxonomy. So this gene may change. One plant gene now you can submit for a particular character, and next day or another sometimes or 100 years of back it will change. It may be changed, but this will not work. They are telling like that. So now the global DNA barcode, the barcode of DNA barcode for land plants, they are trying in the consortium of biological barcode of life, recommended to use the chloroplast genes. They have now it is coming like that, but they are also working. This group is working for the giving the complete barcode for all life. Next slide, please. Next slide. Yeah, this also already I dealt with the number of species it has names. How barcoding works, it is very simple. Now every day all gardens it has a barcode. So every garden it has a barcode, and simple technology, sensing technology it has come, and if you can just click your mobile, you can get all the information, and no need to because of the DNA technology, and they have given the that sequence and converted into barcode, and you can see how they have given the sequence into the barcode is like that. These are the sequence. You can see that the dark and bind parts. So now every plant you can fix the barcode is like that. If you are just you get to the gene sequence, you can change into the gene sequence into barcode is very simple, and that can be used for that identification tool. And another one recent innovation in this technology. Next slide. Go to next slide. Yeah, you can see the QR codes. The barcode may be converted into QR codes. Even now, the famous gardens in India or abroad, every trees you have this kind of QR codes. Even you can do in your college campus or university campus and everywhere. Just we can create one barcode. That barcode may be converted into the QR code for particular characters. That may be depicting in every trees. Just we click it that barcode, you can get the whole information what you have saved in that barcode or you given already in the program. So these are the very simple technology it has come. Even a lot of the free softwares available in the computer, just you can convert into the QR code. The QR code may be getting all the information. So like that, the changing epitome of species identification. So previously orthodox taxonomy, only they are rely on morphology. Now some in between it has come some chemotaxonomy, chemical based identification, or numerical taxonomy. So now is entirely they have turned up on the molecular systematics. So most of the species now, you can see there in this more than the 1 lakhs or 75,000 species that was identified only based on molecular technology or molecular systematics, because already we have the APG 4th classification. So how means it is possible only because they inventing new PCR machines. So now it is mini PCR and several PCR system. Every day they are introducing an advanced system for the extraction of DNA or multiplication of DNA, and also developing parcoding technology. So parcode development also, every day they are inputting some new kind of technology. So these are the things, global genome biodiversity network. Just you will get into the computer, you can see the number of specimen, you can get the information, what are all the species already they have done and what type of the information stored for gene sequence and everything you can get from this site, the global genome biodiversity network. Even almost all the globe it has covered, nearly more than crores of specimen they have already developed barcode. So uses of the DNA barcode, it's a very simple, the research tool for taxonomists. It is one of the research tool, already told you, molecular taxonomy is one of the research tool, but it is not giving for information entire organism or entire globe. So to aid an identification of species, to expand species diagnosis to all life stages, to test the consistency of species definition. This is one advantage. Suppose this tool is available, the morphological identification, we have to wait for flowers and fruit. Suppose in any orthodox taxonomist, if you go to identification, just you have a twig means they asked about is either flowers or fruits are there, because that is only the identification character. Even mere vegetative characters we cannot identify, but these tools will be there, means we can identify any stage of plant. And second one, applied tool uses for taxonomy. It is one of the applied tool, recently originated tool to identify regulated species, including invasive. So we can identify any species, suppose already it has been stored in the information. To test purity and identity of biological product. So already Vijay Shankar Raman has much explained about this, how can we detected the purity and identity of the products. So that identification and authentication important for all materials what we are using in any kind of things for our human purpose. So that can be identified properly. We can use it, one of the technology, barcoding technology also we can use it to assist ecological and field studies of poorly known species. Because then a lot of species are poorly known species, or sometimes it's a very rare, so only confined into the very specific region. So that can be developed for this kind of technology, easily identified and locate for this. From this tool, we can do like this. And third one is discovery tool. To flag potentially new species, especially undescribed and any cryptic species that can be differentiated or maybe described. I have given example also. So finally, we have published a taxonomy of angiosperm textbook for degree student. So we have explained all the modern things very simply and clearly for your in our own language, and we have given the local examples that is more important. And finally, I would like to thank the organizer, particularly Dr. Vijay Bhaskar Reddy, and all the other the organizing committee of the webinar for giving me the platform to discuss things like taxonomy. I'm very happy to be shared my knowledge and in this platform. And once again, thanks to all the organizers and all the participants also, because I think most of the participants are known faces. I saw the names are also, and I'm very, very thankful to you, sir. And once again, thanks to all. Thank you. Any queries and comments, you can send it to my mail. Already I have given my mail ID. You can send it, or else you can ask me now also.
Thank you so much, sir. Very detailed and elaborative session. I think some of the participants would like to interact with you.
Yeah, ready.
Karthik, please take
So
this.
if anyone wants to ask any question, please raise your hand and I can answer.
Wait.
Some people are already in the panel. Nartana, yes.
Sir, I have one doubt.
Yeah, please.
What is the difference between Caralluma adescendens variety fimbriata and Caralluma adescendens variety adescendens?
Yeah.
Could you find any difference between these two, sir? Any
Yes.
small?
Already a lot of differences are there. Caralluma ascendens, Caralluma fimbriata. Now it is in a species level. They have erected in POVO. But
Yes.
previously it was published Caralluma ascendens. 5 varieties are there. The first described by Cravely and Mayuranathan. He is the first monograph of India that was there in I think 1940s. That monograph was there in Caralluma. And the fimbriata, it is not there at ascendant, that attenuate tip. That is a major difference between fimbriata and the ascendants. Ascendance means it's growing like that, that conical. No, it's towards the attenuate tip. That is ascendance. But fimbriata, it is not that attenuate tip. Along the stem, it is having same diameter. That is a meaning of the basic difference between. And fimbriata is having more hairs on the petals. You can see that purple hairs on petals is a very more hairs on the petals. But at ascendants, other varieties not that much, very sparse hairs on the petals. And there are other microscopic differences also there in Polinia and the outer corona. These are the differences are there.
Okay, thank you, sir. Thank you very much.
Welcome, ma'am.
Someone is asking, sir, one more question. Sir, any feature is there for chlorophyll to DNA and amplification?
Please, sir, can you once again question, please?
Yes, sir. Sir, any PCR is there for chlorophyll DNA amplification?
Any PCR we can do. PCR is a common technique, A chlorophyll not specific for chlorophyll or chlorophyll DNA. PCR we can do. There is any kind of DNA we can multiply. Just PCR machine is multiplication of DNA fragment. The isolation is important. So if you ask the question about the isolation technique for chlorophyll DNA, that may be varied. Or extraction of DNA may be varied, but the PCR technique is common for all the molecular techniques.
Megastri garu, you can ask the question. Like unmute your mic and you can ask the question. Megastri.
Thank you, sir. I am Megastri. And sir, my question is, in plant molecular taxonomic study, apart from nuclear DNA, we are using chloroplast and mitochondrial DNA.
Okay.
So among chloroplast and mitochondrial DNA, which one is more stable?
That is a universal one. I told you this RBCL in chloroplast and MATK in mitochondria. That is a stable one. The genes are already, it is the permanent one. Most of the people rely on those two genes only. Chloroplast means chloroplast or mitochondria means in plant system.
Thank you, sir.
Yeah, welcome.
One more question, sir.
Yeah, please.
Any work is done on microscopic and macroscopic characteristics on Caralluma species?
So yeah, already it was there. One man in South Africa, Darrell Blouse, he has published a lot of information on the particular genera Caralluma. He only segregated 17 subgenus subgenera under the same Caralluma species, and he has given a lot of the supportive evidence in micromorphology, in macromorphology, even cytogenetics and chromosomal karyoology, like that lot of information. And later on, Meevan Laid, the two scientists, they have written a book on succulents of the world, succulent plants of the world. And they have listed the special section for the Caralluma group and given all the micromorphology and macromorphology like karyology also for evidence, and they have worked. But in India, we don't have that much work except our work. We have just me and Professor P. Pullaya, we have published the monograph on Caralluma. And we have also included that micromorphology, just we have taken from microscopic pictures and the plates we have put in that book. But not work, the published works, much published works are not there on Caralluma species.
Thank you, sir. Thank you for your insightful information and your presentation. Thank you. Over to Vijay Bhaskar.
Thank you. Thank you.
Thank you once again, sir. And now I request Dr. Sri Lakshmi to propose a vote of thanks. Over to Sri Lakshmi. Dr. Sri Lakshmi, please unmute yourself and turn on your video. Yes.
Thank you, sir. Hello. Sir, it was Karupaswamy sir, it was a great, very informative presentation. I should congratulate you and also thank you giving us a lot of information regarding the rules of nomenclature and the Sensen code, all the details about the usage of the code. It was really lot of information which we never knew that. At present, the scholars, they can incorporate all this information to carry forward the future research in plant taxonomy. And today, whatever information you have given, I really felt surprised that the barcode system is so developed that just sitting before the computer, people can get the things done. So you have given a detailed account of the phylogeny of the plants and also all the different molecular markers, whatever you have given, especially the marker genes, which you mentioned, the RBCL and MATK genes. I'm sure the students will make use of them to generate, to differentiate between different plant species and whatever is the research goal. So thank you, sir. Thank you once again for such an informative session. Also the whatever you have mentioned, the the chloroplast sequence, all the regions you have very, the details you have given are the minute details also you have given. So I'm sure all this will help the research scholars especially and also the PG students to plan their research and the two plant differentiation, that is Caralluma and Bovaceae, both are very well differentiated, and in fact you even elaborately told the advantages and disadvantages of the molecular techniques and also how the barcode system is useful in plant taxonomy. Thank you, sir. Thank you very much. I heard, I know you when I was a student, when I competed for women scientist, DST women scientist competition at Madurai. I have been there. I competed from ICRISAT, groundnut pathology lab. You might be knowing Dr. Hari Sudhini. So I competed from that lab. But now it is really a great honor to me. I should really thank Vijay Bhaskar sir to ask me to give you vote of thanks. It's really a great privilege, and thank you once again, sir, for both the sessions. I would like to thank you. It was very, very informative. And I'm sure all the participants and also students and even other scholars who are viewing your sessions might have enjoyed. It was quite informative. Thank you once again, sir. Thank you very much.
Thank you all.
Thank you, Dr. Sri Lakshmi.
Yeah, thank you, sir.
Yeah, over to Dr. Shashikala, ma'am.
Sir, thank you very much, sir, for once again giving me this opportunity. Sir, Kurupaswamy, sir, actually, you made both so many things very easy, sir. All these days we were thinking that the taxonomy is only for pure botanists, and rest of the people we cannot do anything. But you made one thing, that barcoding and QR coding. So if we work to some extent, then we can do, even non-biologist also we can do something. That information was very useful for people like me, sir. Basically, I am not a pure botanist. I am an environmentalist. I am from environmental science department. So once we made, with our students, we made a survey, like what are all the different plantations which are there in our university. So our students, they did that work, and they want to label. Actually, they want to label with what names of the trees and plants. But actually, we were thinking that even this barcoding can also be given. If we give barcoding also, that will be very useful. But to our limitations, we could not do so, sir. But now, with your this lecture, we can even try the barcoding. We can go to sequencing, and afterwards generating that barcoding. Even this QR code can also be generated. This information, this idea, actually we were not knowing. Rest of the botany people, they might be knowing, but it is very, very useful to even to us, sir. Thank you very much for throwing lot of light in this area, sir. Definitely we are in requirement of such taxonomists like you all, sir. Actually, all the persons, resource persons, are very, very resourceful, and we realized actually how the importance of these taxonomists even in traditional medicines and all those things, because with the identification, proper identification only, we can make different extractions or different medicines. So a lot of information is being grabbed from this session, sir. Thank you very, very much, sir. Thank you very much.
Thank you very much. Even though you can not only get barcode development for identification of trees or plants, not only from the molecular technology, but you can do morphology also. Just you can develop the characters. Characters can be converted into the barcode. Barcode may be converted into the QR code.
Oh,
they were not
really?
No need to that molecular technology for QR code development. That is very simple.
Okay.
From the morphological character itself, just photographs of the particular tree, you can develop barcode. That barcode may be fixed in the tree. Everybody, you will give them some baseline information. Behind the barcode, they will just click means get all the information. You can give all the information for every tree. It's very simple. Barcode development and QR code development is very simple. Even if you give the assignment or that project for your students, they will do very happily and very simply, because they know how the technologies. They have developed much. Already they have a technology. Simply they can do at least 2 trees for one student. They will happily do that things, and it will come. The total things, it will come like that.
Okay, sir. Thank you, sir. Even this information is also new to me, sir, especially to me. Thank you very much, sir. Thank you.
Thank you, ma'am.
Thank you, sir.
Sir, our participants are requesting for PPT slides. If you permit us, we will share with our
Yeah, you can share them.
Thank you so much. Thank you so much. That would be great.
Thank you, sir. Thank you, sir.
Thank you. Dr. Nageshwar Amanchi.
Sir, Dr. Vijay Bhaskar Reddy, sir. Sir, these presentations are whether available in the YouTube after these sessions also?
We will try to, madam. We will try to.
Please, because again, 15 minutes or 20 minutes, sometimes we lose the talk. So I wanted to hear.
Madam, they will be available, madam. Don't worry, madam. They will be available forever.
Okay, thank you very much, sir. Thank you. Thank you.
Over to Srujan. Any instructions for participants?
Sir, you please reflect on the assignments part, sir, because some people are again asking.
Yeah. Please don't worry about assignment, what we have given yesterday. It's a simple thing. Those are sent, it's okay. Those are not sent, you ignore. And already we have received your messages. That itself enough. And we will try to arrange one session on key preparation. Don't worry. Thank you so much for your cooperation.
Sujan?
Yeah, next session will start sharply by 2 o'clock. Please join us back. Dr. Upendra sir is there with us for the second session. Dr. Ramakrishna sir will take the first session 2 to 3 p.m. Kindly get back to us. Thank you.
Thank you, Srijan. Thank you, Karthik.
Thank you, sir. I am ending the session, sir.
Yes.
Thanks.